Můžete si vaši válku zatáhnout se.
Vyhnanec. Víc nevidí.
Já tě zřejmě.
Karle, ty to říkal, že ty to chceš vyslat do něčeho? Jo, já jsem tam hodil včera playlist 7.9. Počkej, to tady nejsme, to jsme tady.
Potřebuju tě. Mít sebou. Musíš být silná.
Musím ještě na to přijít. A tady se dá dělat ty playlisty. No, a já jsem tady vyšel
Již brzy.
ale já nevím, jak to...
Jak to dá do toho playlistu? V čase, to znamená, nemělo by se odkládat to řešení. Doporučuji používat senzoring každý den. Jednoduše proto, že funguje. Vyzkoušejte a uvidíte sami.
No, teď už nám jde jakoby stream, jo, prostě, ale přemýšlím, jak říct, aby se to...
Ale možná problém už asi možná pokrát na to přišel.
No, že tam ten stream, ty jsi tam dal nějaký detail, že a
to je podrobnosti.
No, podrobnosti, no, jasně. Jo, ale počkejte, tím pádem bychom to tady měli někde už jako...
No to by mělo být rychlý, ne? To tak playlist.
Já ten playlist je neveřejný.
No, tak tady něco odešlo. Tady ta bedna hraje, ale hrozně slabě, takže prostě ten výstup nebo vstup nefunguje. Ale víte, co mě napadá? Ještě vám teda donést pár krátky a úplně zvlášť ze zvlášť. Ty chrchátka, co mám, tak když jsme to zkumili, tak to bude mít lepší zvuk, ale...
Tak playlist
ty bych mohl donést.
Teď tam byl.
Já ho tam nevidím.
Co chvilkou tam bylo?
A ty bych vám byl schopen
teďka o tom máš
zapojit.
Tady.
No.
Takže jo, takže asi to je ono, jo? Ahojte.
Ahoj, Vašku,
ano?
pro ty pout...
Jo, musíme jít zavolat Petrželové.
Musí se tam volat
jo, já jsem si domluvaný, že zavolám. Potělačku, tak to je to asi všechno. Tak já ještě mám
kdyby se něco podělal, tak mi nevolej.
Dobře. Vyřídím.
To je samozřejmě nejlepší postup.
Tady je takový vrtům. Úplně zvlášť.
Jako... Ono to na tom ono relativně stačilo. Otázka je, jak to bude fungovat ve chvíli, kdy budou v tom Zoomu ještě, ale tou určitostí to podle mě jako... To, že bude monozvuk, nám zase tak moc asi nemá. Že oni stejně jako na tom svém počítači budou mluvit do nějakého mikráku, který bude jeden, takže budou mít stejně mluvostí.
Jo, spíš, že by to líp slyšeli maximálně. No, tam jde spíš to, že ti lidi, co budou tady, že to líp
tím pádem tam máte asi 20 jestli tam.
A já jsem tam teďka dal ten mikrofon, on ho nějakým způsobem tady vypl.
Když tam vlastně podíváte, vypadá 20 minuty. To je věc, kterou vidíme. Nicméně, ten jeden mod je 1023 část, což je docela velké číslo a v té chvíli v podstatě ty molekuly se chovají statisticky a dohromady jako celé je to popsatelné v statistických veličinách.
Jo, to asi není špatné. Tohle je jakoby ten zoom prostě teď.
Toto by byl zoom teoreticky nějaký mluvení toho dotyčného. S tím, že vy si tady nastavíte ještě úroveň. No a teď tady do toho začnu mluvit, protože to jsem nastavil malinko ty. A kdybych s někým mluvil, akorát, že ten dotyčnej 1, 2, 3, teďka záleží, jestli ten dotyčnej vás uslyší na základě jakého mikrofonu vás uslyší. Jo, kdyby byly dotazy nebo něco, jestli budeme puštěné YouTube, anebo jestli budete mít ještě záznam z toho Zoomu. 1, 2, 3. Takhle bych byl slyšet.
No tady to moc hlučí, tak já to neslyším úplně dobře, ale to je jenom proto, že tady všechno hledá. Ale jako ten mikrofon je...
Nad vším. A je to schválně tak nastavené, takže vlastně teoreticky, když ty lidi se budou dotazovat, tak je možný, a nemusíte to opakovat, je to slyšet normálně, protože teď, když se bavíme my dva, tak je to slyšet. Já to ještě klidně můžu stáhnout, jako tady toto.
No, myslím si, že to je nutné.
Jo?
Dobrý. Ale teď ten zvuk můžu teda strumím tady tím.
Ano, přesně.
Can you hear us, Genevieve?
Да-да-да. Главное начать, а потом уже не важно. Пойдем салат бахнем. Я угощаю. Ты платишь, я угощаю. Ты платишь, я тебе деньги перевожу, потому что у тебя каша. Кроме коровы, это не мое. Что за лэптоп такой модный? 100 000 рублей? Я не помню, как у меня было сейчас. А откуда такие деньги?
Počkej, Karla, možná když to tam vypadnul projektor. Ti vypadnul projektor.
Мне кажется, после того, как я платишь,
Právě, jak se do toho začne
Can you hear us now?
ну, пока мне
Yes, yes I can.
Okay. So that seems that this is working, and only the thing I need to set up is okay. So in that case, see you in about an hour.
Yeah. Thanks.
Thanks.
Online test bude nějak potom prostě v Google formulářích. Google. Jo.
Počkej, už to je.
Nevěřila by se, že to stojí. Takže nějaký 5 na 6.
Dobrá, takže já myslím, že to prostě jo, 12, 15 začíná. Ne, to si a víš, že to běží, ale už jako nás slyší. Takže se to nespoň.
Jo, to dělá Zoom někdy.
Ale my ten Zoom, jako ten Zoom nemáme.
So, welcome here in Olomouc at the 7th Advanced in Silico Drug Design workshop, which we are collaborating and cooperating together with Pavel Poliščuk, who is sitting here as well. And we are organizing this series of workshops for our students and internationally, as I said, for the 7th time. And we are changing the times and the speakers according to the wishes of the previous participants. So at the end of the course, when I will be sending you the diplomas and also the link for the evaluation, please indicate to us what we did good and what we should do better. As you can see from the slide, we have plenty of lecturers from multiple places. We will have 2 speakers from industry, and we have plenty of speakers from academia in Czech Republic and Austria and EBI. And this all event is free for charge because we got funding from free resources, and that's from Aurora University Alliance, where University of Federico and the second in Napoli is, and that's the last institution which I don't have here. Oh, my bad, sorry. And via the Elixir and Eatris infrastructures in Czech Republic. So hopefully this starts to work soon enough. So Palacký University, as you might not know, is the second oldest university in Czech Republic. It was established 1573, so last year we had quite a nice event for 450 years. And as you will see during this workshop, it is really a secret jewel of Central Europe, as the Lonely Planet Guide tells us. And one of the people who studied here was Johann Gregor Mendel, but for some reason he made all of his later genetics foundation in Brno. Okay, so the next point is all the important points for the lecture. So here is main train station. Here is the faculty where we are now. Tomorrow we are going for a conference dinner at the Svatováclavský pivovar, Svatováclav Brewery, in the city center, close to the rectorate of the university. And on Wednesday there will be excursion to the facilities of IMTM, Institute of Molecular and Translation Medicine, at the Faculty of Medicine and Dentistry. And Catherine. So, as you can see, everything is walkable, so that's not a big deal. So that's the reason why tomorrow, when we will finish with the tutorials, we'll go a little bit through the city center, and because it's quite nice. Today, apart from this introduction, you will hear a little bit about the drug design in general, about the problem of drug design and its definition. Then we will have 2 online guests from PDBe in Europe, Genevieve Evans and Paulyne Magana, who will be speaking about what is possible to do with the protein data bank and how it can help, especially together with the PDBe knowledge base. Then we will have a guest from Charles University, which will be Marian Novotný, who will be speaking about the binding site identification. And we will finish the lectures with Alžběta Tuerková, who is already here, about the insights from Biotech, and she will be speaking about the molecular glues Protex that this company is involved in. And then we will have poster session, which, as you might notice, it's just outside of this room. On Tuesday, we will have, in general, the small molecules oriented lectures about virtual screening, quantum mechanics in drug design, and artificial intelligence in drug design. Then after lunch, we will have Johannes Kirchmeier from University of Vienna speaking about the frequent hitters, and then Wim de Haen from University of Chemical Technology in Prague speaking about QSAR and then a tutorial about it. On Wednesday, we will move towards the structure-based drug design, so we will speak about how to obtain the structures of proteins of machine, which I've called alpha foldology, because upon arrival of AlphaFold in 2020, quite a lot of things changed in the field of structural bioinformatics, and now basically if you have any sequence, you basically have any structure nowadays. And then we will switch for Federica Moraca from University of Federico II in Neapol, who will be speaking about molecular drug design and molecular docking together with tutorial. And Alexandra Ivanova from Pavel Poliščuk Group will be having a tutorial about molecular dynamics simulations. On Thursday, it's all on Pavel, mainly, because he will have plenty of lectures there about de novo and how to use that, and about the cage competition, where he was one of the finalists. And his team was, I think it was second or third place? Third place. Okay, well, first place, out of many. Cool. And he will be speaking about that as well. And then for the afternoon, we will have our honored guest, Thierry Langer from University of Vienna, who will be speaking about the pharmacophores and having a tutorial about it. Unfortunately, this year, because of this building being closed on Friday, we had to cancel the hackathon challenge. So, next time. Okay. All the materials will be uploaded to the webpage during the workshop, as I will receive them from the lecturers. So, as far now, there are 4 of them for today, and there are 2 even for Wednesday and Tuesday. Everything, as you can see, all the electronics around us is because we are directly streaming and uploading to YouTube channel, so it will be available afterwards at the YouTube channel as well. So you can even look at that later on. And if you have any questions, then ask them. If you want, well, yeah.
I was really interested in performing the challenge, looking forward to it, and unfortunately it's not taking place this year. But could you please share, for example, the objective of the challenge, so maybe we could at least try to do it without any, competition or something?
Pavel, that's up to you.
Yes. So because I organize this challenge every time, actually it would be possible because we have websites of previous challenges. So we can provide these links and you can use the old data. The objective function is very simple. So we provide a set of ligands which is active or inactive for a particular target, and provide a large set of ligands which are not labeled. And there are just a few hits hidden inside, which is not so easy to discover by searching databases. So I try to find them from different sources. And your role will be just to create your models, any kind of models, pharmacophore, docking, or QSAR models, just retrieve these hits. And when you create a submission list, you can upload it on the website, and it returns you the hit rate. So it is organized in this way. I think this is pretty interesting, funny part of this workshop. Unfortunately, this year it is not be able to provide, but we can provide you all links from all those previous challenges. So we will upload this on the website.
Okay.
Thank you very much.
Yeah, you're welcome.
See?
Maybe what was the success rate when this challenge took place there? So how many people succeeded in correctly identifying those?
Almost all always, almost all groups find at least one key. Hit rates highly dependent on the data sets we choose, because sometimes we are not so nicely hidden these active molecules, so it can be up to 60% sometimes. Last time, as I remember, the winners had something like 30% hit rate. It's not bad. So it's very dependent, very dependent. The range is very wide.
Okay, so this year, instead of challenge, for the students who need this for finishing the exam, I will be preparing the online test that I will be sharing after the workshop, based on the lectures and tutorials that will be given. So for that reason, I don't have it yet because I don't have all the lectures. So I wasn't able yet to prepare the online test. But I think it will be on Google Doc, in Google Forms, so it will be a relatively simple one.
So is the score included on the certificate, or?
Yeah. Well, the certificates will. I usually prepare multiple types of certificates depending on whether you've presented a poster or whether you've applied for, whether you've went into the challenge or whether you go for the test this year. So depending on what you will do, I will prepare the certificate for that specific purpose. Okay, if that's it all, I think you can enjoy the workshop, and
I hope you really enjoy this workshop, find connections, friends, so just relax, listen lectures, and do not be so shy, and you can communicate with any lecturer who is okay.
okay, this is still not working for some reason. So, let's then switch to the first lecture, which happens to be mine, and it's about the drug design introduction. And I like to start with this motto from David Young, whose book on computational drug design is quite nice read, actually, and I suggest you to find it somewhere in the library and to read it, because even though it's quite old now, it still gives you the idea about how the drug design can help with the product design and with drug design. And the motto of this book is that the pharmaceutical company utilizing computational drug design is like organic chemistry utilizing nucleic magnetic resonance. It won't solve all your problems, but it's much better off if you are with it than without it. And ask organic chemists what they do when they don't have access to NMR. And it's possible to overcome this, that you don't have the characterization by NMR. But it's usually not that simple. So the outline of this lecture will be where we are basically taking the drugs these days. Then I will be speaking about drug design problem, and I'll be specifically looking in some parts of it. Then I will be speaking a little bit about the drug targets and what drug targets we have now, and a little bit about the small differences between drug designing strategies for small molecules and biologicals. So the sources of drugs. The first drug was this one, which is morphine, and it was founded in 1806, and it was used basically as a painkiller for everything. So, for instance, the painkiller for small babies not to cry, for general public. Imagine doing this with morphine these days. Somehow we've moved a little bit without toxicology as well. And we are not doing that to our kids, even though sometimes it would be helpful, I would say. Oh, never mind. So, from the ancient times, basically the people were looking for the drugs all over what they were able to find in around them. So, they've taken the plants, they've taken the poisons, and diluted them, as the Paracelsus famous quote about that the drug and the toxin is only changed by the amount of the drug and the amount of the material. Minerals, basically all natural sources. And then we started to find out what are the molecules behind that. So that's the morphine was first extracted drug in 1806. Then it was another chemicals, pigments, synthetics, and it's moved more and more to high throughput and to looking into larger and larger libraries of compounds to be tested. And now we of course start with the in silico screening for potential molecules which does not yet exist, and basically we are building them from fragments as well. So now the amount of molecules which we can find out and then that can be synthesizable is enormous. And basically to find out what compound out of that will be that drug is not that simple, and I hope you will see during this workshop how this works. Another thing which is quite important to note is what was the testing subjects. From the beginning it was humans. It was curing them or killing them, basically no matter what. Then they found out that it's better if they kill someone else, so they tested the drugs on prisoners. And of course the sailors as well, because for instance the effect of vitamin C was first found on sailors in the first clinical trial, which the doctor curing the scaries among the sailors basically was giving different treatments. Among them, I think it was mercury as one of them. Another one was the lemons, and the last one was just beer. And he was looking what happened to that sailors who were given what, and somehow we are not using mercury to cure scaries any longer. So, but testing on humans is the last step these days, and we basically, since we've moved to more of high throughput things, quite a lot of those testing can be dangerous. So for that reason we moved to animals, then we moved to organs, then we moved to cell cultures, and then we moved to even more in vitro, just enzymes and stuff, and onto the chips and recombinant proteins and so on. So we are moving to have more high throughput screenings, but as you will see later, the amount of the drugs which we have per year is relatively stable. So the sources of the drugs, according to this Journal of Natural Products paper, is from 30% synthetics, but the rest, approximately 50%, is coming from biology. It's either a natural product or some derivative of natural product, which makes sense because we are part of nature as well, and therefore it's useful to use the compounds that are belonging to nature as well. But since we are at the beginning of this, I think I should speak a little bit about the vocabulary which we will be using. The first part is about target. Because usually these days, when you try to find out how to cure the drug, you have to have a mechanism of action. And mechanism of actions quite oftenly requires that you have a target onto which you are heading. And target can be any biomolecule which is interacting with your drug. Then the drug stems from the lead, because you are selecting the compound and then you are optimizing it. And the general motif you are optimizing is called lead. Because we are using the screens and we can have multiple compounds which seems to be useful, we are calling those hits. because they are above some kind of a threshold, and therefore they are hitting it. And from the hits, we are basically selecting the lead. And we are selecting candidate compounds for the next round of testing. There are two possible ways how to look at it, at the activity. And that's the efficacy, which is qualitative. And it says whether we are curing or not. So it's just binary option. But binary is not always the good thing, because we usually want to have some kind of a concentration dependence of how big the effect is. And for that reason, we have the activity, which is quantitative property. And that allows us for the selection of the dosage. So depending on how strongly your drug binds to the target, it quite changes amount of the drugs which you have to take in order to be active. So for instance, for the acetaminophen, Paralen, or paracetamol, the typical dosage is 500 micrograms, yeah? Milligrams. And that's because this drug is actually not that much effective. Because it's relatively small molecule, and for that reason, you have to use quite a high dosage. It also comes to the effect that basically the drugs typically don't interact just with one target, but they quite oftenly have some off-target targets, let's say, which can lead to some side effects and so on. So, for instance, for paracetamol, the side effect can be a liver damage. Okay? So you have to take care about the right dosage for that specific patient, and it quite depends on how strongly the drug binds to the target and how strongly it binds to anything else.
And we are talking about painkillers.
Yeah.
What about maybe aspirin or acetylsalicylic acid, which is of course an SAD, but it binds to its target like irreversibly. So it stays on its target. So does that make difference too, since it does that irreversibly?
Yep.
While ibuprofen, which is effective in lower doses, albeit like it's like half the dose, ibuprofen like 200 milligrams, while aspirin is like 500 milligrams, while ibuprofen does that reversibly. So it can swap places and go maybe to another target.
The major difference there will be that the cell will need to make the receptor again to make the protein again, and for that reason, it quite changes the longiness of the effect, basically, and the resistance to it.
And follow up: is there some case or is there some logic where to select if you want to design an irreversible inhibitor, for example, or a reversible one? Does it matter on the target, for example, or it doesn't really matter?
It does changes kinetics, pharmacokinetics a lot, and in some cases can change a lot. Also, the effectivity of the dosage.
Yeah, but there is no general rule that, for example,
I'm not aware about any, but it might be. Yeah. But I'm not an expert for covalent binding drugs as well.
Chemical warfare agents are examples of a lot of irreversible.
That's true. Yeah.
Neurotoxins.
Yeah, that's true. Because if you want to kill someone, it's better to do it irreversibly.
And maybe also protection against the warfare agents, because if there is some protective drug and it's reversible, then it might be outcompeted by some poison. But if it's irreversible, it might protect for some point.
Actually, I've heard about this in one grant application and in one project that they were basically trying to come up with some reversibly binding drug, that would basically be able to detect the warhead before it will reach the target molecule and interact with it earlier than whether it will be interacting with this acetylcholine esterase. But I don't know how it went, and I don't think that I will be able to find it in the literature because it was done with the OEA, Department of Army. Okay, so the last term is bioavailability, because it might be that you have quite a nice drug or nice hit that seems to be binding perfectly and interacting nicely, but it can come up from the in vitro study. And when you try to work with the cells, cell cultures, or even with organs or animals, it might not be effective because it's possible that the compound will not be able to interact, will not be able to basically get to the place where it will have the active. And that's unfortunately quite often a problem, because the drugs by the time are bigger and bigger, because they tend to be more selective. But if you are increasing the molecule, then of course you are changing the permeability properties, and therefore the drug is not that available to go through to the target if the target is inside the cell. So it has to be balanced in some way. That's at the end the activity of the drug in vitro does not necessarily translate into effect when you are putting that to the patient. And that's actually one of the major points why the drug development fails these days.
Is there a case, maybe stupid question,
yep,
that the affinity or the binding would be too strong and some person would optimize it in the other way, that they would like to the drug to bind less?
I've never heard about the case of that, but because that would change the dosage. Yeah.
Yeah, exactly.
So I'm not sure, because, for instance, if I took the anti-HIV drugs, which were developed at the IOCB, the first versions of those drugs were effective, but only at a large dosage. So basically the patient had to take the pills of almost 10 grams every day. And that's quite a huge amount of the drug. And of course, at that amount, it also had quite a lot of side effects. But during the development, they basically were able to tune it in the way that basically the latest one, I think Truvada, you are taking just one pill per day, and it's relatively reasonable size. So usually it's the other way around than just from the really strong binder. So once you have the drug, I'm just comparing here the conventional pathway of vaccine of development of the drug, where you start with the preclinical research. Then you start, once this is successful and you have the compound which seems to be effective, then you go for phase 1 for the safety to find out what is the safe amount that you can give to the patients which doesn't have the disease. Then you go for hundreds to see safety and immunogenicity. And in the last one, you basically are looking for strong enough results based on the dosage you've selected, whether it's really effective. And then you get the registration. You can see that this whole time is approximately 10 years. And on the left side, I have the individual in silico methods that can help with within the selection and within different parts. For instance, in biology, to select the target, that's the analysis of GWAS. Then the finding actives via QSAR, pharmacophore, or de novo design, and then evaluation optimization, you can use docking and molecular dynamics. But we all lived through the COVID era, so it somehow did not take that long for development of COVID vaccines. And for that reason, it was because multiple phases were done in together. Because they've combined individual phases quite quickly one after another, and it was relatively easy to go through the last phase, where you need the largest amount of patients, because there was some kind of a pandemic. So you had plenty of patients, and therefore you quite quickly can see whether your treatment have any effectivity against the placebo. So really quickly, it was visible that the vaccines are active, and for that reason, we got this provisional registration, and now all of them are actually fully registered. But this acceleration was basically accelerated because it was necessary and it was really in need at that time. But there are multiple other ways how you can speed up the development, and that's, for instance, if you have a rare disease where you don't have many of patients in individual countries, but you can have relatively reasonable amount of the patients all over the world. And for instance, quite a lot of those diseases can be quite problematic for those people, and it would be really helpful for them if they would have some kind of treatment. But to develop drug for only 1,000 patients will not pay off if you would go through all the process. So for the development or orphans, it's actually a little bit easier to get the registration. And the next drug development is then usually going that, okay, you have orphan drug, and you know the mode of action, and therefore you are then trying to find out whether your rare drug can have effect on something else, on something similar, or let's say some similar cancer variation. And then you are moving with the drug also to try out some new illnesses and so on. And it's quite interesting to look into how the registration changed during the times for the individual drugs. Also, if there is nothing for that specific illness, then it's also easier for the registration because we are first in class. So we are the first one to basically offer something. And so on. So, how we are on it? I just yesterday updated this graph from the last year. And while last year I was quite pessimistic about going downsized in both cases in European market and US market, because this is by Food and Drug Administration, this is EMA, European Medicinal Agency. You can see that the amount of drugs per year is approximately 50 in US, in average, for the last, let's say, 10 years, and approximately 40 in case of Europe, and that's when the last year was basically a relatively good one.
Is there a reason why US market has more approved drugs than European market?
Basically, from what I've understood when I was reading those, there were quite a lot of generics here as well. But I'm not including the generics in here as well, because in that case it will be 75. Okay. So maybe that's because US is paying more, and then only you take from the US and register it as well, and in that case, we are basically registering as a new thing, which is not that new, so maybe that's the reason why we are basically not the first ones. And another point, which is unfortunately true for biomedicine in Europe, is that every country is fragmented. We don't have, when we are doing some kind of a research in biomedicine, for instance, in the hospital here in Olomouc, usually it's quite hard to include the patients into studies which are abroad. And it's quite complicated to select the patients and then to combine them with other countries. And it's much more harder than in the US, where they don't have that much of the protection of privacy as we have with GDPR, and with medicine it's even worse for the research.
Since Europe is a larger market, and from what I've
But it's more fragmented in this case.
Yes, and from my very anecdotal evidence, actually most of the drugs are like, it has been approved here in India, but these drugs are not approved in US market, since you have US, I thought it was more conservative, but now I'm proved wrong.
Doesn't seem that way. Another thing is that approximately 50% of those drugs are for orphan diseases. So that might be the reason why you are not that much aware, because rare diseases usually don't come into unless you have it. So, or some of your family. So that's the reason why the amount of the drugs is higher than it was earlier, when there were no orphans at the beginning. So this orphan drug development is one of the way how to generate more drugs which are really needed for those patients. I was quite surprised that last year expedited, therefore quicker drug development was actually lower than any previous years till 2018. So the US market slowed down a little in this. In Europe, it basically fluctuates about 30%, so it's also probably a little bit slower here. And you might be familiar with Moore's law. And Moore's law is about the speed of computers, and that it's quicker and quicker every 2 years. You basically have double speed and so on. In the development of drugs, we have Arum's law, which is Moore's on backwards, and that's the number of dollars investment per drug. And you can see that the drugs, that this line is slower and slower. So you have to have more and more money in order to get the same amount of drugs. But you see that this graph ends in 2010. So I moved further, and I found that there is a recent OECD book about artificial intelligence in science, which at one page analyzed the Arum's law as well in pharmaceutical development. And you can see that if you add the recent years, it seems that we stopped the Arum's law for some reason. And the major reason for that is that we are moving more and more into innovation, that now it's relatively cheap to do the high throughput screenings. And we are better now with producing the relevant screening methods and models. So that's quite optimistic, I would say, from the point of Arum's law. Yeah.
What about inflation? Because if it stops in this graph and there is inflation, then it's actually declining.
It is. Yeah. That was the another one which I didn't put here. So if you look into that book, you will see that for the investment, it's still not that great.
So we need to be better. Then there is another myth, and that's the missing novelty in drug development. And it was analyzed 2 years ago in the review of financial studies. And this is the evidence that the risk aversion leads to underinvestment in innovation. This is the Tanimoto coefficient, which when you compare the fingerprints of two drugs, basically how the drugs look like, you get the one value which will give you the idea how identical they are. So at 1, this is identical drug. And if you are around here, that means that you don't have anything similar in the previous drugs. So this graph tells us that in general, there are 2 peaks. One peak is finding something new, and one peak is finding something completely the same. That's quite strange, isn't it? One thing, the peak which is about the same is the peak for deuterated drugs. Because if you do the deuteration of the drug, the skeleton will be the same, but since you are changing the chemistry around, so you are changing the hydrogens for deuterium, you have a new molecule. So it's possible to go further with these deuterated molecules, and it's one way how to go over the patents. Yeah. So that's this peak. The another peak is basically trying to find out the similarity and to find what's going on with the new things. And here I have just a selection of statins from the 87 to 91 and 91 December. And the first one had the similarity score 0.525, so it was something like here. And that was the new molecule. The newer one was Pravachol, and because there was Mevacor, it's basically here in 0.6, and Zocor was 0.8. So basically in here one, this part of the graph is giving us information about Me Too drugs. Because quite often the pharma companies are developing the drug for the same illness, which are similar to one which is already at the market, but it's somehow changed. And for that reason, but the general part of the molecule might be the same. So this part here are Me Too drugs. Quite a lot of those. One thing which is quite funny is that the larger companies which have more than 20 drugs are actually more likely to engage in novel drug development rather than smaller companies with smaller amount of drugs. And which is from this graph. So if there is the company with previously 0, the new drug will be less likely to come. And if we compare this part of the graph, which basically those Me Too drugs, towards the proportion of the FDA approved, the highest FDA approved are those ones which are quite similar, but you don't get that much money from those because you basically have already competitor in the Me Too drugs. And the drugs which are completely novel can come up with the largest patent value in money. So it's still quite often the innovation of a really new drug can lead to highest income afterwards. But it's not that easy, and usually you need quite a lot of expertise, and more expertise is usually given in a larger companies. So, the most typical mechanism of action, which is coming from Emil Fischer, a Nobel laureate, and he said that basically it's like a lock and key, which you are basically the target with biomolecule, and the key is your drug. And he was specifically speaking about the silver bullet interacting with this target. But finding this target is so-called drug design problem. And the identification of new drug is quite expensive and quite hard as well. Because as you've seen, the expenditures per a new drug requires more and more money. And that was just for the R&D, not for the production, not for the patents, not for the distribution, which adds more to the costs. And the new drugs are then quite expensive. And it's also a hard problem because to find out the good pair between the target and drug is not really simple. And there are the admit properties and side effects and so on. And possible obstacles are also that you might not have a testing model. Because, for instance, HIV is a human immunodeficiency virus. And even though we've tried a lot of humanized mice, still not to my knowledge, we have one that would be useful as a typical model for HIV development. Not one. So that basically slows you down because you can't have the animal model to test. And it's not really ethical to try on people. Then, for rare diseases, as I've already said, the amount of the participants, the people who have the disease, might be quite small and might not be able to pay for full price of drug development. Fortunately, since 2000, let's say, 8 or 10, I'm not completely sure when exactly was the date when these orphan drugs started to be allowed to the market, to have expedited procedure and cheaper procedure. And now, as you've seen, it makes 50% of drug development. Another thing which is quite common is that you find new drug which is less toxic than the previous one, but it's also less active. And that's quite often. That basically new drug which is developed is more safe and so on. But unfortunately, quite often it's less active than the previous ones which we already have. So it's quite problematic. And we already talked about Me Too drugs. Another point is that not all illnesses are the same. When you have enzyme overproduction, which is case for some cancer types, because in cancer the cells are growing and dividing, and basically if you want to stop the cancer, you basically stop the division. And for that we are using inhibition, kinase inhibitors for instance, and those ones is really typically small molecule strongly binding towards the target, towards the kinase, and that's it. The problem is, quite often, it doesn't bind only to one kinase, but it quite often binds to a series of kinases, and therefore, once you are targeting to one specific, you are actually shooting shotgun to quite a lot of others, and therefore you are generating quite toxic compound which can stop something else. Then you can have a higher response of some receptor. So for instance, COX receptor in pain, and for that you can have some kind of antagonist which is binding towards the COX receptor and stopping the activity, as in pain relievers. Okay, again, small molecule binding towards the molecule, but in this case you have to select the inactive form of the receptor. Another thing is, and this one is harder, if you have a low response of a receptor, so you don't have enough of signal coming through. And for that part, these are ones of the hardest one because you have to basically increase the signal of something, not to block it. Therefore, you have to basically put some kind of gain of action, gain of function towards the drug. And it's more complicated than blocking something, because it's not about just about the binding, but you have to bind specifically as an agonist, and more strongly than the natural ligands of that specific receptor. But we are doing that for neurological diseases with neurological GPCR proteins, and for instance, serotonin receptor agonists. Then it can be not, another possibility might be that the regulation is done via the peptide. One possibility for this is CGRP peptide in migraine. And if that is wrong, it's really, then you have to come up with some kind of a peptoid drug that mimics that specific peptide, but or you can come up with something that will basically blocks the peptide, let's say antibodies, blocking those peptides.
It's like the case of semaglutide. It is now pretty
yep.
widely discussed.
Mm-hmm. Yeah. And there is emerging field not to focus only on proteins, but also to focus on RNA. And it's quite interesting field as well, because all the CRISPR biotechnologies are basically the RNA interactions. So there's a huge amount of possibilities, and each of those possibilities basically guides then what methods can be used for that specific protein. Then we can look into what are the targets of the current drugs, just to learn where we are shooting it. So this is the graph which I made in 2016 to our review in pharmacology research, where we were specifically looking for membrane-bound proteins, and we were interested into whether those membrane proteins are actually important for drug as a drug targets. And I was quite surprised when I've looked into Campbell database, and I was just looking for the targets of the drugs which were approved, and I was quite surprised that quite a lot of those targets were either membrane receptors, typically GPCRs, or enzymes which were on the membrane, or ion channels. So 60% of the proteins are actually membrane bound, and this is actually the point where AlphaFold helped the most, the structural biology, because those were underrepresented structures in protein data bank, because previously it was quite hard to get a protein structure in membrane. Nowadays, with AlphaFold, it's relatively easy to get a model of membrane protein and use this as for a prospective finding of the drugs. The rest are basically transcription factors, which makes sense because you are changing the way how the cell interacts to a chemical signal, so how much it will change the protein expression. Another enzymes directly binding to the DNA, transporters, enzymes in cytoplasm, and there is even a membrane itself somewhere. Yeah, 0.5 in top. Which is, for instance, antibiotics. One route of antibiotics is basically attacking the membrane of the bacteria itself.
Is there a difference of the AlphaFold structure's quality, which can greatly influence the success rate of successful small molecule design? If you would compare it to X-ray structures and AlphaFold, how big is the difference?
I will be speaking about that on Thursday, on Wednesday morning at my AlphaFoldology lecture. But it seems that there are two differences. The first one is that AlphaFold seems to provide more hollow epo structures. So it seems that it will not be that useful as the PDBs with the ligand. But in a recent study by Skojcet, they've used prospective drug design development. So they basically selected based on the screen on the AlphaFold models or on the PDBs. And they've stated in their Nature paper that they come up with completely different sets of molecules, but when they test it, they get pretty same hit rate. So that was quite surprising result for me, because previously all of the previous versions were comparing the docking towards the PD versus PDB, and for that retrospective screens, AlphaFold was really not useful. But for this prospective one, it seems that it might be worthwhile. It was quite surprising result, I would say. So, speaking of Campbell, it is quite nice database by the by the EBI group, currently under Barbara Zdražil. And one thing which is quite funny when you look into the drug targets there, and you compare them with the actual drug targets, you can see that in some cases the drug development is much larger than the amount of actual drugs coming out of that. So, for instance, here are GPCRs, and you can see that basically the level of GPCR drug development and the actual success of drugs coming from that is quite, I would say, similar, but a little bit more. The more of the drugs are actually there. But if we look for proteases and kinases, there is much more compounds which were analyzed than the actual drugs. And if we compare the amounts of these as some kind of measure of the intensity of research, you can say that it would fit here as on the second place in total. But unfortunately, the success rate in those places is much more limited. And as can be seen here, the GPCRs are also the part where the largest amount of the innovation is nowadays driven. In case of kinases, it's quite smaller amounts, and only in the recent years it started to grow a little. So, I'm coming to the last part, and that's a little bit different when we compare a small molecule with biologics. On the left, we have aspirin, which we've already spoken about. And on the right, that's a monoclonal antibody. And it's from the size perspective, you can see that we are looking at completely different beasts. And you have to use completely different methods in order to analyze small molecules such as aspirin. Typically, we wouldn't be that small these days about small molecules, molnupiravir being the different, because that's the latest, smallest drug I've seen. But we are approximately in a range of, let's say, 400 to 600, 700 Daltons. But monoclonal antibodies, which are quite often parts of the production, are much more often. Are much bigger, and therefore you cannot tune them that specifically, and you basically rely on some kind of a preparative stasis. But as our logo of 7ADD shows, the last letter D I've just made when I was trying to make a 3D print for this, and I wasn't able to find some nice protein native one which would resemble the letter D. There is a solution in alpha foldology because there is a method called protein MPNM, and this method basically allows to generate any protein sequence that will fold into a given shape. So I've took this and it's available in collab on GitHub. So basically I've selected the letter D. Based on the sizes, I've selected the number of amino acids, push the button, actually 2 buttons, because first you have to install everything in collab and then you can run it. So 2 buttons, and it generated me 3D structure, which when I've printed it, I think quite resembles letter D. So there are different methods to do that these days. And when we looked into those new molecules from the last year, we actually see that 34 of those were small molecules, not much of peptides or radio-labeled. That's usually, from what I've seen, it's usually 1 per year is one radio-labeled one, and the peptides can be here. Last year it was basically the same in 2022. And then we have the biologics, proteins, and here typically the antibodies are generating the majority. Then there are some bispecifics, enzymes, and by 1 for fusion protein, and here it was just 2. And then the last year there were some nucleic acids as well, more than it was a year ago. There was just 1 cRNA a year ago. So all of those changes, all of those modalities are now being approved. So can be, as a take-home message, drugs comes from various sources. It's hard and expensive, but thankfully for us, due to biology, not due to the computational problems. It's somehow we have to mimic those, so okay. And actually, one thing which is costing more than our development by in silico and everything is clinical trial. It still is much more money than the development in preclinical stage. Clinical trials basically trumps everything. The most typical drug targets, as I've said, are GPCRs, ion channels, nuclear receptors, and kinases. But there is a long tail of small or those orphans, and those orphans are making a huge part of the accepted new drugs. Biologicals are more complex to produce and to train, and unfortunately I think that we won't have much about their development in this workshop. But there is a workshop at Brno, at Loschmidt Laboratories, about the protein engineering, and that's more relevant to that. And the last part is the most important. There is no gold path. There is not one path that has to be taken. All methods has to be tuned for a specific project you are working on.
How about the drugs like semaglutide that are peptide-based? Are they in category of biologicals or category of small molecules? Since they are not small molecules, it's a peptide, but they are produced like, yeah.
Peptide. Yeah. Small molecules. Still. Because those ones which are in biologicals, that's really a full protein. So that's much bigger thing. And it also comes through a quite different metabolism and everything. So it has to be dealt with quite differently.
Yeah, that was like my question, because whether it's classified by the size of the molecule or the path of synthesis, since these peptides are synthesized. I think semaglutide is synthesized on solid state, solid phase.
Yeah, because it's smaller. So yeah, typically the biologicals comes from the biologics, that you have the cells that are producing it, and yeah. Okay.
I got one question.
Yeah.
You know, how is it modified by drug repurposing?
Drug repurposing, well, that's basically what I was speaking when I was speaking about how you have one specific target for your drug and then you are moving to another targets and so on. And that's this hopping to a new illnesses is basically part of the drug repurposing, and it's quite valid to look for that as well. But if we remember those basically the COVID drug development, the first drug which was available in the market and which is working was remdesivir, and that was drug repurposed from, and because it was previously developed for the African Ebola here. But then the ones which are now used in clinics, mainly before you go to the clinics, that's molnupiravir and Paxlovid, and both of them are new ones. I wouldn't go there.
Yeah, but the pathway is interesting.
Pathway is interesting, but unfortunately it does not seem to play a role. So,
Nirmatrelvir is inhibitor of...
Yes, of one of the proteases. Yeah, yeah, that's Paxlovid. Nirmatrelvir.
Paxlovid has two components, doesn't it?
Yes, together nirmatrelvir together with ritonavir. But you use the ritonavir as a some kind of a how to shut down the cytochrome P450s in order not to take down your peptide-like drug.
But the best one to work with is remdesivir in the lab.
In the lab, yes.
I'm using it. It is the most efficient, the best control.
Yeah. But I must say that with remdesivir, I like that it's prodrug of the prodrug of prodrug.
Best control is very good, you know.
Yeah. But I like that it's quite nice way how to, because if we came up with the molecule which will be active, and that comes to the question, maybe your question about the molecule which is strongly binding, and we are making it less binding, we are actually doing it. If I came up to this, because we are quite oftenly producing prodrugs. And prodrug is not binding that efficiently, but it has to be turned on by the metabolism inside us. Yeah. So for instance, ibuprofen and apoibuprofen, which is basically ethylated or methylated ibuprofen, which first has to be cleaved by this, and then the ibuprofen itself, which is the active molecule, interacts with the COX receptor. So yeah, we are doing it. Okay, so that was all from me. Sorry that I was too late. And now we'll switch for the... Okay, and now we'll switch for the Paulyne and Genevieve. So, hi.
Hello.
Hello.
So I'll share my screen.
Yep.
And yeah, so coming to you from the UK.
Okay. Can you hear us?
I can hear you. Might have to, if there's questions, I might need you to repeat the question, but that's okay. So thank you for inviting me to participate in your in silico drug design challenge/workshop. And I'm presenting about the protein database, which I've already heard referred to. PDBe came up a couple of times in the previous presentation, was mentioned. So I'm based at PDBe, the protein bank in Europe, and I am a structural biologist EBI, look for our training materials, look at the training materials for the PDBe. And in addition, we also have a YouTube channel, and you can go to that YouTube channel and go for a walkthrough talking about our pages and also both our pages that are entry focused, but also our aggregated or knowledge-based pages. And I'll sort of distinguish those a little bit as I go through this presentation. And today I'm going to try and do a bit around application programming interface, or APIs. And there's quite an extensive webinar, combination of videos, tutorials, notebooks that are available with this link here. And there's also a start page that sort of orientates you. And so I just wanted to share that. So we're going to touch on that a little bit today. So hopefully, can I just get maybe a show of hands in the room? Have people been able to access the survey and respond?
Yeah.
Wonderful. Oh, that's really excellent. Fantastic. Okay. So, what is a PDB? So a PDB is actually one of the older databases. So it's 50 years old. And it is an archive of three-dimensional structures of biological molecules that have been experimentally determined. All the structures in the PDB have been determined using experimental methods. And it covers proteins, peptides, and proteins in complex of peptides, proteins in complex of DNA, proteins in complex of RNA, RNA in complex of DNA, DNA in complex of DNA, and of course gamma ligands to all of these various things. So there's a lot of different macromolecular structures that are present in the PDB. And the size range is quite large. So you can go from really small peptide-like things that bind, like this copper scavenger I'm showing here on the screen, and I'm showing that in stick representation, whereas in sphere representation, not to scale compared to it, I'm showing a Zika virus, which is 4 megadalton by comparison. So 4 megadalton versus less than 1 kilodalton. So very large ranges of size. A key aspect of the PDB database is this thing called the PDB code. And this is something you'll often find if you're looking at a structural biology paper or a paper where they've done drug design and they've been able to capture the drug binding to the target or the hit or the lead binding to the target. They'll have listed near the end of the paper in their data availability section a set of codes, and those codes will enable you to go and view the three-dimensional data at the PDB. Every time a structural biologist publishes a new structure, they have to deposit their data with the PDB. We also have data that hasn't been published as well. Sometimes structural biologists will upload their data, but they won't be able to necessarily publish it. So we have data that also corresponds to published work as well. And I think I'm going to mention here, so this is about 2 years away now. So we've been going for 2 years, and we're essentially about to run out of the alphabet. So in about 2 years' time, we're actually going to change the format of our IDs, our PDB ID codes, so that we can have more codes and we won't run out, because we're about to run out of the 4-letter ones. And I just put that link there just in case this is something you're all interested in or want to be aware of. There's more information there about how we need to change our codes. A key thing about our database is that the data is three-dimensional. That means you really want to be able to rotate it, spin it, explore it in 3 dimensions. You want to sort of be able to go in and out of it. And so Molestar is an amazing viewer that we have implemented on all our web pages, which can help you navigate that. Another key thing that's really important to know if you're interested in small molecules is that in PDB terms, we tend to call small molecules ligands, because they're usually in the context of something being bound to a protein, an RNA, or a DNA target. Here, there's a heme group bound to this small protein, and the way in which we handle ligands is they also have a unique ID. So we have the unique IDs for the structure, for the individual structures, the individual sets, the experimentally solved structures, and then we also have unique IDs corresponding to the chemicals. And so I've got the PDB CCD, so the chemical component ID there for aspirin, so it's AIN. If you wanted to find all the structures that contain aspirin, then this ID would help you find them. So every time there's a structure solved with aspirin present, they have to use that CCD ID to define it. And again, we're about to run out of the alphabet, so we are in the process now of start making IDs that are cloud-related characters as well as the free character ones. We refer to these as chemical component IDs, which may or may not be a helpful term to bear in mind. We have PDBe-KEM pages, where you can essentially plug in an ID or plug in a name, and you will find all the corresponding records related to that chemical. And then from there, you can use it as a launchpad to find all of the structures which contain that chemical. An aspect to this kind of data is because we're trying to describe in 3 dimensions these large, complicated macromolecules, they're much larger than a small chemical, that we have to describe them in terms of a hierarchy. So we describe the atom positions in 3 dimensions, we give them a sort of couple of things that kind of correspond to error or / uncertainty. And then in addition to that, we have to describe how those atoms come together to form either like a ligand, so like ATP or aspirin or something, or how they come together to form an amino acid. And if they're coming together to form an amino acid or a nucleotide, we then have to describe them in the context of a polymeric chain. And because of this nature of trying to capture all of this information, we essentially have tables that look like this in our data. Now this is what a standard PDB files look like. And if you notice, I'm sort of trying to highlight here, there's kind of a grid aspect to it. In terms of, you've got the residue names, you've got the chain names, you've got how the residues sit. So residue, I should say, is PDB talk for an amino acid. We talk about them as being residues in the context of a polymer. And then of course we have to have the atom. The atoms have names assigned to them, as well as the element is defined over here as well. So this was each atom is described in a position in 3 dimensions. And in addition, occupancy is a way of describing the presence. So if there is a ligand that's only, well, I can get to that a bit more later, but essentially the data is a summing up of multiple instances of the macromolecule in the context of the ligand. And if only half of the macromolecules have a ligand down and half of them don't, then the occupancy here will be half. And similarly, we have something called the temperature factor, and that relates to some extent to the uncertainty of which we have with the position of the atom. This is particularly relevant for X-ray crystallography. Structures determined by X-ray crystallography. It's a less useful metric than some of the other methods because it's not as standardized how that this particular column gets used. So the thing I should mention is it is this kind of table shape. The previous picture here is a legacy format that we've used for many, many years. But it's not the current way in which we hold data on the database, and we actually just generate this for the community to use. And the data is actually held in a different format called EMBL-EBI. And this is the master format of the PDB database. So when you go to the PDBe, you get an option to download the structure. Often the first option, because you're trying to convince more people to use it, is the EMBL-EBI format. It's still a text file. It's still got all of that information relating residues and action positions and occupancy. But it's got additional metadata in there, and it's actually much more robust in terms of data storage and computer passability. And I just wanted to highlight 2 things here, which is one is that EMBL-EBI is well described with lots of different software involved in using it. Here at this website here, but also some software refinement, software sort of giants in terms of structural biology. CCP4 and Global Phasing have got together and made a really powerful parser that can enable doing all sorts of tricks with these type of files. So if you're interested and you want to learn more and you think that you're going to do a lot in structural biology, I highly recommend get on the EMBL-EBI train, as it were, now, and start to learn about how this file format might be able to empower some of the research you might do. So either an EMBL-EBI file or a PDB file can be equally viewed in visualization software. So I've mentioned Molestar because it's embedded on our web pages, but there are a lot of other visualization softwares out there. So and most of them are freely available. You don't have to pay for them, or you can get an education version of them. So there's PyMol, Chimera X. There's also one that structural biologists tend to use when they're building a structure, which is Qt. And there's other options out there which you can try. Molstar has a web browser, so you just have to type in molstar.org and you'll be taken to a web page where you can just actually just type in a PDB code in the viewer and you'll pull in that structure and you'll be able to rotate it around and view it in 3 dimensions. So why have a central archive? Why do we have a central archive? Well, to some extent, we inherited it because there were some far-sighted scientists about 50 years ago who thought this was going to be a good idea. It is incredibly hard for us as a structural biology community to have this database. And it really enables us to query the quality of the data because we can validate our structures against all other structures. So this butterfly-shaped structure over here is actually a structure I saw quite a while ago when I was doing my PhD. And so I deposited it on the PDB. Now I wouldn't be able to dig through like notebooks and find information about the structure anymore, but because I've deposited it on the PDB, anyone in the future that ever interested in the structure, they can go and look at that data and they can analyze it. And over here we've got this analysis over here which shows that I actually did quite a good job in terms of, so these different metrics tell us something about the quality of the structure and the position. So these positions correspond to what is happening in my structure, and where they're sitting in this bar corresponds to how similar they are to other structures in the database. And so that capacity to validate essentially structure against other structures that have already been solved is one of the things that enables us to have good quality data. Also, in terms of, we can consider the structures in context of either in silico models or other types of data, and we can also consider the structures in terms of components themselves. So we've got I'm just showing icons here of some ways in which we are looking at those structures in the context of things. And one of the great things is that it's a worldwide database. So all the structural biologists in the world deposit their data to the same database. And that's something else I want to mention is this is a coordinated effort. So it's not just one site. We have sites around the world. So essentially when a structural biologist deposits a structure, they go to one web page, but behind the scenes, depending on where they are in the world, it gets sent to a different site, and then we all use the same software and the same processes and policies to process their data. And then every week, we release an updated version of our database with additional data that corresponds to the latest set of publications that have been made available. And you often won't even realize that you're looking at data from the PDB, because you'll be looking at somewhere like GLASS or Uniport or PubChem, and you might see out of the corner of your eye a macromolecular structure with some things bound. It's syncing with our archive. We are the holders of the archive, and we sync with everything else. And an aspect to bear in mind is that people have downloaded that data and done really wonderful things with it. Obviously, one of the things that you're going to hear more about later on this week is AlphaFold. So AlphaFold trained itself on these 200,000 structures that the structural biology community had been depositing into a shared archive for about 50 years. And that's what helped them to have parameters and to be able to train and then develop new capacity because they were training on this data set. And similarly, there's another thing called null property. They downloaded a cohort of data set of structures that were really high quality, and they used analysis of that high quality data set to then have quality metrics to judge other data, and potentially even to be incorporated into pipelines that you might be doing in silico design type stuff. You might be using some of that analysis in order to interpret whether you've got a good model or not. So, as I mentioned, this is a collective effort. It's global. It's happening all over the world. There's people working. So I'm at the PDBe. There's also the RCSB, which is the Persian data bank in essentially North America. We've also got PDBe. PDB China is a partner of PDBe and associate member, or becoming an associate member. We've also got, there it is. I don't know, you can't see the list, excuse me. But we've also got PMRB, which is specifically to support structural biology, where NMR has been used as a technique to solve the structure, and EMBL-EBI, which is a database to support cryo electron microscopy. Because that kind of data cohort is a little bit different, there's additional database is to support that data. And so there are actually quite a few experimental techniques that we support and we accept, but the primary ones that get used to determine three-dimensional structures are X-ray crystallography, solution NMR spectroscopy, and electron microscopy. And there's recently been huge development in how powerful electron microscopy is, and therefore, if you look at the numbers, you can see they're just comparing like ago, only 10% of the structures being deposited in the PDB were electron microscopy structures, or structures determined by electron microscopy, and now we're moving towards 30%, and maybe next year it'll be, moving towards up towards 40%. So last year we actually passed the 200,000 milestone, and as of today we're at 215,000 plus structures. So just to touch a little bit on the data that's supporting, so although what you might download from the PDB is primarily three-dimensional atom descriptions of a macromolecule, the actual data that's supporting it is not the three-dimensional structure. That model is an interpretation of experimental data. And in the case of X-ray crystallography, that data is called an electron density map, and that map is essentially taking a crystalline sample, putting it into an X-ray beam. The diffraction is then sort of, you imagine a microscope, the light goes out, that's the diffraction, and in the case of X-ray crystallography, we use mathematics as a lens to focus it back in, so that we can then have a map that we can interpret. And so the model gets built iteratively into this map, and one of the things with this particular technique is the more you build, the bigger your map becomes. You want prior electron microscopy, you'll have a grid, and your sample gets put on the grid. There'll be plunging, and it gets plunged prior, it gets frozen, and then multiple instances of that small molecule are determined. That's actually something I should mention here. Is in a crystalline sample that you're getting your structure determined from, there are multiple instances of the macromolecule. So this is a summation of multiple instances of a macromolecule bound to your small molecule. And a similar thing happens with EM. EM is a summation of multiple instances with slightly different orientations, and it generates something called an EM volume map, and that is what the model gets built into. Similarly, nuclear magnetic resonance, it's a summation of a solution of macromolecules bound to ligands, and then chemical shifts are determined and analyzed, and the strengths are built, and you get. In the case of solution NMR, you end up having multiple models because of the way in which the data works, the way in which the restraints are taken into consideration. Oh, and so just to highlight here, this is the database that's dedicated to supporting EM data, and BMRB is the database dedicated to supporting NMR data. So, continuing on, what's in the PDB? So I mentioned PDBs, but obviously there's proteins, and in the previous presentation, a lot of the targets that were indicated, so the ion channels, GPCRs, and kinases, they're all proteins. But another thing to be aware of is we also have nucleic acids, and nucleic acids might be, potentially, some of the drug targets in the future, because they are involved in gene regulation. Some of them, in fact, have roles in catalysis, there's ribosomes. A lot of drug targets, some of the drugs that are being deposited in the PDB in terms of the context of a macromolecule is a ribosome bound with small molecule bound, and they think the small molecule is targeting the ribosome. The ribosome is a combination of protein and RNA and DNA and all sorts. So sometimes you might be considering a protein or a drug target in the context of quite a complex system. So the nucleic acids have secondary structure, they have a structure that can be considered, and so these sort of paddles here represent individual nucleotides, and obviously double helix is quite a famous secondary structure that we're aware of in macromolecules and the understanding of three-dimensional macromolecules. And here, I've got a few more unusual structures here, including this one called the quadruplex, where the strand actually sort of twists around on itself. Now with proteins, you may have seen lots of cartoon and representations of proteins, and there are actually many ways in which you can represent a protein. And you may have seen this curly structure and not necessarily know what it means, but essentially it's a way of describing the backbone. So if you think as a chemist, and you think about the R groups, and if you imagine removing all of the R groups and substitutions on something that has a standardized scaffold, that's sort of the way in which we think about thinking about the amino acid, the peptide bond that's happening in the back, there are these hydrogen bond patterns that can happen. And so alpha helix is one particular hydrogen bond pattern, and I'm trying to show it there. And obviously you can have multiple alpha helixes, and as it's flashing through, so you've got this tip representation, and then it should flash through, highlighting those hydrogen bonds, and then it flashes through to the alpha helix. And then another way, of course, you can look at these is space filled, where you're looking at all of the atoms and sort of a Gaussian or some sort of surface representation, solvent accessibility, that sort of analysis. And we also have beta strands, which again, they're this arrow, we draw them as this cartoon arrow, but it is a way of representing a way in which the backbone of the protein is folding in on itself. And if we move on from just thinking about the secondary structure, there's also the tertiary structure, and the tertiary structure is a way in which we describe how a secondary structure, so you can imagine if you have a string of beads, and there is the amino acid sequence, and then the next layer up is the secondary structure where there's alpha helices, where there's bead sheets. There's actually a couple of other types of secondary structure that I'm not touching on, but those are the main ones that we think about. Then the next step up is how these things come together and form together into a shape. And really that is what creates the binding pocket. It's if you just look at a protein sequence, you don't really have a good sense of the binding pocket which a drug target might bind. It's only once the protein folds up into its three-dimensional shape that you start to see that binding pocket. And there's other interactions involved, like salt bridge, disulfide bonds, that are often critical for forming that three-dimensional shape. Or actually coordinating the ligand with a cofactor or a metal might also be critical for forming that three-dimensional shape. So we have a search feature called PDBe-fold that can help you find similar proteins that have similar tertiary structure. And in addition to that, if you're interested, if you've got a target, and maybe there isn't a structure for it currently, but maybe there's something where there's a whole bunch of structures with something closely related, to help you find that, you might want to look at something like Intipro, where you're looking at protein families or protein domains, and you can group together related information in order to get more insight. In addition, obviously, is, I sort of alluded to this with the ribosome, is that you can have quite complicated combinations of proteins and ligands and nucleotides, forming these essentially multiple polymer chains that fold together in a tertiary structure, and they come together, or you can envision them coming together to form this higher order structure that involves multiple chains. And they can be relatively simple, or they can be really huge, like the ribosome, and often it's mediated by interactions between hormones on the side chains, hydrophobic patches, or salt bridges, or disulfide bridges. And we have a tool at PDBe called PDBe-KB that can help you understand and analyze those interfaces and the assemblies. So, one of the aspects of the PDBe is if you just type PDBe into Google or whatever, it should take you to our webpage. And there's a couple of different search bars. I just wanted to quickly highlight that there's this advanced search feature. If you click on that, you'll come up with tons of options of which you can group together potentially a complex of experimental structures that might be relevant to your drug design. So I just wanted to highlight this because I touched on it before. If your target, for example, is a bromo domain, because bromo domains are quite popular, they're found in quite a lot of part of quite a lot of different proteins which might be considered drug targets. If you type in bromo domain, or if you type in the ID that is given to bromo domain, you can get a whole set, so 552 structures contain a particular type of bromo domain. And so another thing I would mention is that with the PDBe website, what you don't want to do is treat it like Google, where you partially type a word and press enter. You want to partially type a word, see what options pop up, and then click on the option. That will give you the most benefit from using this interface. And so I think this is time now where I'd like you to either using my Bitly or using the GitHub, if people can connect to the notebook, and I'm going to quickly run through, it might not be so quickly, we're going to work through a notebook that I prepared, talking through how you can supercharge or, I guess, leverage, I think that's the word I use in another book, leverage of APIs in order to get more information about the PDB. So as I say, 200,000 plus structures. It might be hard to find the particular correlate of information that's most relevant to your project, and so that's why APIs might be beneficial. If the tutorial I'm presenting is like too simple for you, I redirect you to this GitHub because they've got more advanced notebooks. This is a somewhat simple one that I'm going to lead you through today. It will hopefully just give you a taster of how this kind of code works, how this code might work for you. Yeah, so maybe I'll just give it a pause now and ask, are there any questions at this stage from the things that I've presented so far?
Thank you. Do you hear us?
Yeah, I can hear you.
Okay, thank you. So some questions from the audience before they start this tutorial part of PDB. Are there any? Yes, please.
I would like to ask what happens to the PDBs of papers who don't get published at all eventually.
They still get released. So the option for the scientists is they can let us know that they either want to withdraw their data, or we actually, once a year has happened, a year has passed between them depositing the data with us, unless they tell us they want to withdraw it, we release it, and we release it without a publication. Because we actually capture so much metadata about the experiment and about the model that even without the paper, that data is still valuable to the community.
Thank you very much.
Yeah, please.
And I don't actually know what the number is in terms of structures we have that aren't published. It could also be structures we have when we haven't found the publication. We do search for the publications, so sometimes the structures we might not quite find which publication they belong to.
Okay, thank you.
Hi. It has been a long time I didn't hear from you, Genevieve. Yeah. Is here?
Use the microphone, yes.
Okay.
Directed microphone.
Oh yeah, from this side.
Yeah.
So first thing is that I'm pleased to hear you, and then I need to make some apology. Then I also can say that I couldn't recognize your voice because you mix different kind of accents. Sometimes there is Australian gang there, and there is a question where is I hope it is not. I got a question. I have one entry, maybe, which is with the group I worked in Singapore, Malaysia, where we submitted some heme binding protein from thermophilic bacteria. So, and I have one question, and it is not dehydropiconylate synthase. It is about one misnomer, which is under the number I. Do you have access to entry?
So you're talking about a specific deposition?
Yes.
Okay, so as it was indicated before, it actually gets split to different geographic regions. So if you've deposited a structure in Malaysia, it's probably being handled by the PBJ team, which means I have no access to it.
Okay. There is one entry which I like you to clarify, and it is with ID IPR 05364.
I'm sorry, I missed that part of the question.
What do you want to clarify with this?
The functional protein.
But, so, what is your explicit question?
The question is that I will answer it. The name of the protein is ermine, and it is obviously misnomer, which is some sort of cytoskeleton binding protein of brain. I mean, it would be better if it was some oxidoreductase in wine that would make, or some flowers, rather than they abuse my name for such dodgy protein, you know.
Okay, so the naming of proteins on the PB, we sync with Unicode, and so we try and name consistent with what happens with Unicode. So if you have an issue with the protein name as if you contact Unicode.
I'm teasing you, okay? Okay. Yeah, thank you very much. Nice to hear from you.
Thank
Right.
you. Other questions?
And, while the question being asked, can people in the room put their hands up if they've been able to find their way to a notebook on an API basis? Okay, looks like some people have anyway. That's a good sign. Okay.
Okay, so we can start the part of tutorial.
Okay. So let me just write it down right first. So hopefully you've been able to make your way here to my GitHub. And just in case you haven't, I'll quickly show you how to access. So you can click on the notebook. And we're going to open it with Colab. So just click this icon called Open in Colab. And what I'm going to do at first is just demo through the front end, and then there's an exercise at the end that I would like you to try. So as I was saying, this is a notebook about APIs, which are a programmatic access points. And I've got some details here about how you can use one of these notebooks. If you're using it on Colab, you need to have a Google account. If for whatever reason you don't have a Google account, you can also download the notebook and use it, either look at it in a browser using a Digital Lab instance, or you can even look at it, download it and play with it offline and do whatever you like to it. It's just a notebook full of cells which contain code and cells which contain words. In this particular notebook, we're running through this resource that we have at the PDBe, which is these API endpoints. And I don't know how many of you have used a notebook before, but there's this button here. The play button here. If you see it, it appears when you mouse over. And essentially, if you click on that button, then you run whatever code is in that block. And so in this case, I've got it all sort of laid out here, but essentially, as I press play here, I want you to run, I'm employing some Python packages that I'm going to use as part of the code that I'm doing, and I'm also defining some variables to find the API endpoints. And I'm going to start with a really basic example. And as I say, if you've got more experience, I suggest you work by yourself on the more advanced notebooks that I indicated. So this basic example, I'm just going to talk through what sort of data structures we have in Python. And in particular, this valuable data structure called the Python dictionary is really useful when you're working with APIs. So a dictionary is essentially has keys and values. So in this really simple example of the dictionary I have here, PDBe-ID is a key. The value is this particular PDBe-ID. There's another key here, which is the experimental method, and the value is the method, which in this case is electron microscopy. And in order to tell Python that this is a dictionary, I've got these two curly brackets. And essentially, when I run this code, not only do I define this dictionary, but I'm also running a few functions that I've written here, which are able to look at this block of information, and based on me telling which key has pulled out a different set of information. So I can tell it this key, and then it pulls out the information of the ID. I tell it this key, and it pulls out the information of the electron microscope. And then I've got this, so these are just directly, really simple code directly getting out of the dictionary, and then I've developed this small little definition, which handles the fact that the key isn't there, essentially returns that there's an error, and it's not, the value isn't there. So moving on to the next section here. So here, as I say, we're looking at this entry. If you go on here, I'm going to get distracted. You can see what the structure is. So this is an ion channel that's an interesting target. So this is, the structure we're looking at is actually the ion channel which nicotine binds to. And we're using it as a template. only part of this web page that you can interact with the PDBe is behind the scenes these JSON files. And so this is a version of it taken offline. And essentially we can convert this JSON file into a dictionary. And then again with some simple Python codes. So this is, I'm using a code called, where is it here? Pretty print. Ah, here it is. So I've got this pretty print here function, and this converts this somewhat ugly looking thing into this slightly nicer looking thing. And then I have a couple of other functions that just analyze the dictionary and put those in more detail. But essentially, we find out that it is confirming that it is a dictionary. In this case, there's only one key, and the key is the ID. And then we've got these sort of indented information, which is like dictionaries and dictionaries and dictionaries. So it's kind of nested dictionaries happening. And so in this particular object that I've created, there's only one key. What I'm going to show in this next section here is we can go down to the next layer. We can use this function here, essentially, to get down to the next layer and create that as a dictionary. And when I create that as a dictionary, then I end up with 13 keys corresponding to values that we might be interested in, like the assemblies involved, the experimental method, maybe related structures, release date, things off the title, of course, that would be relevant. And then, so just showing that a similar kind of, this more complex looking object, I can use very similar commands to essentially do this. You tell the value, tell the key and get the value. So it's actually relatively simple. If you've not seen Python code before, it might seem a bit intense. So just again, one step further is that I can have a little bit more code, and then I can essentially analyze and create an output, analyzing what's in those values, and make this sort of summary statement saying the entry is titled blah blah blah, it was released on this date, and it was determined using electron microscopy. So this is a relatively simple combination. If you imagine accessing data, you can make this quite complex and quite relevant to a particular project, potentially quite fast. So all I've done so far in this, so just to show you here, in this section 2 is I've just gone through some of the basics. And now we'll switch to actually doing API calls. So using API call, we're going to use a package called request. And then we're going to use a package called read. Essentially, the package called read, we're going to do some reg expression matching so that we can make sure that what we feed into the system, into our request, is in fact the correct format for our PDBe ID. And that's what this here is, and this here. So I've got some code there to check that when someone uses this function, whatever they're telling here is the correct ID. And so if I run this, there's the definition. Down below is me using, essentially what I've defined. And if I give it a code like this isn't a correct code, or if I give it a code like this isn't a correct code, and my function tells me that essentially it's an invalid PDBe code. It's an invalid PDBe code. But if I give it a correct code, like the one which is the target of nicotine, the structure of nicotine bound, then I get all sorts of information. And this is coming live on our website, of our servers. And then of course I can generate again a summary. So I can then create essentially do a bunch of summaries. And this is again picking up our website, pulling up information, and creating little summary statements about structures that are available on our website. So far I've been looking at one of the API endpoints, which is the summary endpoint. We have another endpoint, which is the experiment endpoint. And so again, this gives slightly different JSON file. This was the first JSON file. This is the second JSON file. These both come from, I think, the same structure. Yeah, 8CAU. And so again, I can, I forgot to run something, let me see. What did I forgot to run? Yeah, so that's one of the things we should mention is that with notebooks, it's always good to make sure you run the blocks in order, because usually there's some independency as you're going through. So using this code here, I've essentially, again, got the information, and then I've been able to pull out and make a little statement saying the resolution of this structure is X. And I can do the same thing here, and actually create combined the statements. Oh, sorry, wait a second. Oh, I see. This one is a structure that's an electron microscopy structure, and you can see there's some details that are particular to like the microscope. These are all related to the microscope. And then this is an experiment for an X-surface lithography structure, and so it has things like crystal work, which I wouldn't expect to see in not an X-surface structure. Yeah, so some things related to that. And once you have essentially your definitions in place, you can then do queries where you query multiple things at the same time to get a concatenation of information. And so at this point, I just wanted to highlight in terms of, I chose resolution in particular as something to query because I think it's important, especially if you're looking at structures that are either electron microscopy or X-surface structures, to be aware that the resolution of your structure might be quite relevant in determining how confident you can be in the way the living has been modeled in the binding site. So resolution is essentially, so scientists always strive for the best resolution they can get, and there are many things that can impact, and maybe they weren't, the sample just meant there was never a possibility of getting a better resolution for the structure. And a key thing to know about the resolution is the smaller the number, the better the resolution, and the more confident you can be on the accuracy and the precision of the atoms in the model. Another thing to be aware of is solution in the margin, because it's a very different technique. It is, well, I should say it's very different. It is different in a way that means that resolution is just not possible. It's not a measured data quality. It doesn't exist if you have a solution in our structure, so don't go looking for it. Use other metrics to decide the quality of the data. So I've just got a summary here going through some of the different functions I made, or we made together since you're running the notebook alongside me. And I've got some hints here if you wanted to build your own robot and have a play with our API inputs. And then I've got this exercise here. I just wanted to give you some time to go through the exercise. Maybe, I don't know, apparently there's some people online, I'm not sure if breakout groups are an option, or yeah, well maybe just in the room itself, if you can group yourself into maybe either pairs or 3 people together to help yourself help work through this exercise here. I think maybe I'll give you 10 minutes to give it a go, and then we can come back around and sort of we'll see how people are going. I haven't had a chance yet to look at what you responded in terms of your experience with programming, so I'll just have a look now. Hopefully, it's what I presented so far isn't too intimidating. And if there's any questions, or please don't hesitate to ask. And so if you can go through exercise 8 at the end of this notebook and have a go, essentially querying PDBe's APIs. So I'll hand over to you guys, and yeah, please put up your hands, I guess. That's the easiest one, if you've got a question.
Yeah, thank you, thank you. So you can start these exercises, and if you have any questions, you can ask. Up to 10 minutes. If you will finish, you can notify that you're already finished with this example.
can I just ask, how are people going? Can I maybe have a show of hands of how many people have been able to finish the exercise?
Some of them already finished, but not all, as I understood. Maybe just a couple few minutes.
Okay, cool.
One question, when we search for something, there should be a pop-up. I'm not seeing a pop-up on the search bar. It looks a bit different than in the screenshot.
Did you hear?
Sorry, I didn't hear that. Could you repeat the question, please?
Should I repeat it or should I get a microphone? Sorry.
Yeah, microphone is there.
Okay.
You can get closer a little bit to ask a question.
Yeah. So we instructed to specify some detailed options in a pop-up. I'm just not seeing one, and what I'm seeing in my browser looks a bit different than the screenshot in the instructions provided. What am I missing out on?
That is a good question. Let me just show you on the screen. So if you go to the PDBe and you start typing nicotine.
Yeah. So it's before I press enter. Okay.
Yes, don't press enter. So
Okay.
that is the trick with the
Okay.
PDBe
Is this?
changes.
Okay.
Is the way our underpinning structure is not the same as Google. And so we're really powerful search features, but they're all coming through essentially pop-up options.
Okay. I just got too ahead of myself, and I got lost in the filter results and the advanced search functions with already a list of search results present on the screen. Thank you.
No problem at all. And so you can click on that, and then in downloads, this is the one that I suggested to, just in terms of it's an easy file to trim out so that you only have the information that you need to do an API call. Obviously, there's a lot of other options. And if you wanted to, you could repeat the exercise with, I'm going to spell that screen. Cheat and put CC to LG in. But yeah, there you go. So you could also do the equivalent thing with something like aspirin, download, pull it across, run the same kind of structure. But this is just a taster, obviously.
people completed, but probably not all.
Well, maybe I'll just run through a demo with the second sheet while other people are completing.
Yeah.
Just so that, so you've been working through this basic sheet. In the basic sheet, essentially I'm doing get API calls, but there's also post API calls, and if you're familiar with APIs, which I think quite a few of you are, you're probably familiar with that distinction. And that's quite a good thing to be aware of when you're making API calls with the PDBe. And I also want to share, so this is one of our pages that can guide you through some of the API calls. We've actually got quite a few different ways in which you can do API calls. I'm going through the entry-based APIs, but we've also got aggregated API and Solr-based query system API, and you can aggregate all sorts of data out of our database using these different API endpoints. And so here, essentially, the way this page is meant to be a web page that can help you just explore APIs, all the APIs we have. And so we've got, this is a get call, and this is a post call. So the get call, you only have a single PDBe, the post call, you can have multiple PDBes. And so my second Colab, which you can work through in your own time, essentially does get calls instead of, post calls to get. So it's working with lists of PDBes. And I'm just going to be a bit of a cheat. I'm going to run all of my code at the same time. That just all runs through. Again, I've got some headers here to move through. And so the code's running, making some functions. Some of the code in here I have is just to try and deal with potentially accidentally entering the wrong code or the wrong file structure. So maybe dealing with both lists and strings of data and still being able to get the data retrieved, no matter how you're implementing it. And so yeah, lots of different things here. But the key thing I wanted to highlight from this page, so you can see obviously pulling a lot more data, a lot more of the post, but is the fact that we also have, let's go back here, these ones are focused on structures, but we also have API calls that are focused on those CCIDs, so getting all of the information associated with a ligand ID. And so in this case, essentially, I got the ligand IDs corresponding to a bunch of structures. And then essentially here is another set of definitions where I did a call with a list of IDs and got back smile strings. So this is just a taster. I'm sure you could build much more exciting and more relevant to whatever you're interested in, APIs. But I just wanted to highlight the fact that we have both post and get API endpoints, and that you can both come at it from an entry or experiment ID kind of focus, or you can come at it from a small molecule focus. And there's quite extensive information that's available with API calls. So this is just a taster to hopefully make you interested and explore more as relevant to whatever you're interested in. Yeah, has everyone managed to finish the notebook now?
It seems yes.
If you have any questions, there is inputs, PDBe help, and you can send your questions. Just make sure to include the title of the notebook and that you're asking about the notebook. That way I can find your query and respond. Okay, so let's get back to the presentation. So one of the things in is how to pick the best structure for you. How to pick the structure that you might do your molecular docking into, or your in silico analysis of, or the set of structures you're going to do your in silico analysis of. And so how do you tell if a macromolecular structure is good? And I've got 4 basic points here. One is the quality of the data, so for X-ray crystallography and cryo-EM, that's maps, and they make up the majority of the PDB. Another aspect is the resolution, and two more aspects. One is the geometry, and also validation metrics, which is linked to geometry. So in this first section, I'm going to focus on the first 2 points, which is that maps are the real data, and that resolution matters, but it's not everything. And I'm going to focus on this from a small molecule perspective. So this first image I'm showing here is electron density map, so that's maps from X-ray crystallography, and this is what a resolution 3.7 angstrom looks like. If you start to go to 2.4 angstrom, you start to see a little bit more clearly that map is more correlated with the model, with the atom positions in 3 dimensions. And as you go to even lower resolution, increasingly, the map, the actual experimental data, is corresponding very closely with what is modeled. So this is a heme group, heme, so that's bound to hemoglobin and transports oxygen in our blood. So there's, I'll have a chemical structure on there for you. And so one of the ways in which we try and help people navigate the PDB is using Molstar. You can automatically open whenever you're entering an instance, looking at a structure. We have experimental data where the map is shown on top of the model, so you can see how confident you can be that that small molecule that's modeled there actually has experimental data to support it. And small molecules are particularly vulnerable to sometimes part of that small molecule, if it's sitting outside the binding post, so I'll be the show. It's sitting outside the binding post, so you imagine the binding post here and here. It can end up being very flexible, and it might not be very observable, the exact position of how that part of the chemical binds. Or it could just be, it's just not got a lot of interactions in the binding pocket, so it's adopting multiple conformations, and that meant that the experimentalists, when they were examining the data, particularly strongly and confidently model. And so in this instance, this is a protein called fatty acid binding protein. It's a human protein that helps with transport and binding fatty acids. And in this case, this is the same fatty acid bound with the same protein, adopting slightly different conformations. And to be honest, neither of them have the best density to support them. Probably going to be most confident in the region that has the oxygens. And it's a little bit because, especially cryo-electron microscopy, X-ray crystallography, it's the electrons, not the atoms, that are helping, that are doing the diffracting. So it's when the X-ray interact with the electrons that there's diffraction. And so because of that, the more electron-rich an atom is, the more likely you're going to see it on an electron density map when you're doing, looking at maps corresponding to models for X-ray crystallography. The other thing I should mention here is there's 2 types of maps. There's a blue map, which essentially should surround the atoms, and then there's these negative and positive maps, which are green and red. So just to touch a little bit more on what the green and red map. So the blue map corresponds to essentially it's almost reconfirming what's in the model. And so you would expect it to cover, there's a ligand there, you would expect the blue density should hopefully cover it. If the blue density isn't covering it, then probably you have less confidence that that part of the ligand is in that particular position in three-dimensional space. The red map corresponds to an area where there's the amount of atoms that are modeled there is more than what we'd expect. So this red-green map is called a difference map, and it's to highlight where essentially there's a bit of the model in red, that's what is highlighted, it doesn't match the map. And the green is to highlight that there's a bit of area in the map where there's too much map for what's modeled. So there should be more model there to describe the map. And in this particular case, so this is a side chain which wasn't modeled particularly well, all we have to do is reposition the side chain, and then we have the side chain is more correctly matching, or the model of the side chain is more correctly matching what is in the experiment data. And again, just to remind you with resolution, what you're thinking about that number that we're giving is how much we can distinguish between 2 points. So the smaller the number, that means the more we can So with cryo-electron microscopy, it's a little bit different. You've still got resolution, and again, I'm going to go through the same exercise, looking at heme and looking at the different resolutions. So there's a heme at 3.5 angstroms, there's a heme at 2.7 angstroms, there's a heme at 2.2. And to be honest, the electron density is looking pretty similar between them, so it's perhaps less clear. And so one aspect that I want to mention here is that the resolution that is reported for cryo-electron microscopy is so-called global resolution. And global resolution can be different than local resolution. So I'm going to take this example of something that's a potential drug target, an ion channel, a ligated ion channel. So this is the same kind of, in the same protein family that nicotine binds to. And so I've got the EM volume maps there on, they're there for you to have a look at, so you can see the raw data. And if you go to the publication, they report both the global resolution, so that's the resolution we have in our archive, but they also report local resolution. And so you can imagine if your small molecule is bound somewhere in here, you've actually probably got a map resolution that's better than 3.4 angstrom because it's more like 3 angstrom, matching this color. But if you're in this structure and your ligand is down here, and the global resolution is 4.1, but this part of the map is actually more like 5 angstrom, then you have a lot less confidence, potentially, in the way that your small molecule is oriented relative to your protein or binding target. So it's really important to turn on and / pay attention to the experimental data. And that's really easy if you go to our web pages because we load it for you instantly. So you can quickly go and have a look. You've seen a paper, there's an interesting small molecule that you're really interested in, it's bound to a target that you think is great for drug development. Go look at the data, go see if the experimental data supports what has been modeled in the structure, or how strongly it supports it. Maybe it supports it, but maybe there's potential ambiguous and ambiguous. There's something ambiguous, potentially, about the way it's orientated that you might need to be mindful of. And so I want to take some more time to do the other tutorial that I have here, and this is a tutorial to guide you through using Molestar, and particularly to look at how you can view small molecules on our pages. And so I've got a bit.ly link here that you can use to access it. You can also just go to the GitHub and look at the PDF. And this is going to take you for our web page. I've chosen a structure from a recent paper, which is so this structure is of a clinical, it's a drug that's in clinical trial stage of phase 3. And it's from Bayer. They've published a nice set of structures. Some of the structures, if you look at the paper, it's a really interesting paper to read alongside, going through looking at structures on the PDBe, because they essentially found a good lead, and they took it and did looking for how good it is in the context of animal trials or permeability, all of the other factors related to developing something that's good in terms of the good qualities for a potential drug. And they found some of their initial ones weren't really good, and they had to go back to the drawing board and try different algorithms on the same scaffold to try and get the same kind of binding in their target protein. So I'm going to let you maybe I'll let you guys go for 10 minutes, and we'll see how people are going after 10 minutes. And please ask any questions if you get stuck. If I was in the room, I'll be walking around and asking you how you're going, but I'm here, so I can't. So please feel free to put up a hand and ask a question. Cool, okay.
Thank you, sir. You have 10 minutes.
If you don't finish it in 10 minutes, it's fine, but just so that we have a sense.
How is it going? Is it okay? Is there somebody who is still not complete? Maybe some questions?
Yeah, does anyone have any questions?
Seems no questions.
I'm currently looking at another structure from the same paper.
I'm
a bit disorientated.
My apologies, I might have a question.
Okay, yeah.
The whole Mostar session is computed on a computer that I use, not on some external device, right?
Yeah, it's computed on a computer that you'll use. It's computed within the browser.
Yeah, and have you tested it with multiple browsers, or which browser is the best for Mostar?
So I tend to go between Chrome and Firefox, and I've never noticed a difference.
Yeah. Yeah, that was basically the question, since many developers forget about Firefox and they optimize only for Chromium, but for now I had a pretty good experience with Firefox, so kudos for that.
I mean, I did develop it. It's a very wonderful package slash software, and yeah. So the thing that I don't actually have in the tutorial is, I'm just playing here, but if you just want an image, you can just make a snapshot of whatever you're doing in that browser, and immediately you have a PNG that you can chuck into a presentation. So if you have a particular drug that you're going to talk about, and you know there's a protein structure out there, a structure of the drug bound to the target, and then you can essentially find it in the PDB, load up Molstar, get it how you like it, click that little, the picture button, I guess that sometimes, it kind of looks like the way your camera icon for your camera phone might be, and then essentially you download, you press copy, and you get an image. The other thing that I often play with as well is there's quite a few animation options. If you want to have a spinning structure, that's also an option. So yeah, there's lots of bells and whistles with Mostar.
And if I may add, there is export geometry as well, and that's actually the option which I'm using when I'm preparing this, the material for 3D printing.
Ah, lovely.
Yeah.
Yeah, I haven't used that particular tool yet. I'll have to look into that.
Yeah, please.
Is it possible to do this 3D visualization in Python or Jupyter notebooks, or only in browser?
So it's a Java-based. Java is underselling it really, but it's essentially it's been developed for a browser. So there have been some attempts on Git to make a version of Mostar that works within Python, and so you can have a look there. But yeah, it's better in an HTML type setting at the moment. It's not really been optimized for Python, unfortunately, or Python notebook installation, which is why I didn't have any in my notebooks. Yeah. Yeah, but maybe you can be part of the Git pull and requests to develop a version that's amenable to Python notebooks. Any other questions?
Can I have one? I think it's page 5 of the instructions PDF, or basically on the right-hand side in the control panel, there's the region of the protein, of the ligand, and so on. It's something with focus in brackets. I've been trying out the settings, and this preset or pre-configuration seems to have disappeared. Is there an undo button somewhere?
An undo button?
So basically on the right-hand side, there's three entries and 2 more focuses, essentially. If I understood, it's a predefined selection of what I was working with.
So in terms of Mostar, usually if I want to undo something, I just reload the browser because it will reset back to where it was before. And you said you were on page 5.
Right, yeah. So on one of your screenshots where it says focus region or something?
Oh yeah, my advanced ones, yes.
Yeah. So
yeah, hopefully you can follow my instructions. Yeah, so
Basically, I've reloaded.
There is no undo, but if you just turn a feature on or off, like the eye,
yeah,
you can just turn an eye on or off, and so you can make like a particular visualization disappear for a moment and then have it come back. So I tend to use the eyes rather than the trash cans.
Yeah, I understand it's a hide or show function, but once there's no 3 dots to click on because there's 3 lines in the table where there was 5 previously. I don't know what mistake I've done. Click on the structure.
Oh.
Structure. Because yeah.
Yeah, so the focus target and focus surrounding only appears when you click on the limit, and if you click away, it will disappear, and unfortunately all of the settings you have inputted disappear.
Okay. Thanks.
Yeah. That's a good question, and good work trying to do
so I was
sort of my bonus tasks.
Yeah, I was looking for a selection pane or for a complete list of everything that's being currently visualized, and items just were gone, and I wasn't sure where to look for.
So maybe a show of hands in the past. How many people attempted the optional, additional sort of detailed instructions for visualization that I had, like this one on page 5? See a couple of hands go up. Okay. Good effort. It is actually, if you're trying to analyze a small molecule bound in a structure, there are a lot of things that you can turn on and off in a Molstar instance that can help you navigate. So are people feeling, so I'm going to upload to my GitHub, my repository for this, I'm going to upload a document with the answers. So even if you haven't finished the exercise in this session, there will be a document uploaded with the answers, and you can go through that. And I think probably the trickiest thing I would say from this activity is this instance here. And just to sort of show you a trick, is that you can make it full page, or not full page. And when you make it full page, it's a little bit easier to turn on the help and get the information about what the color coding means. And another thing just to be aware of, as if you notice when you mouse over one of these guys, this arginine here, it highlights over here, and if you mouse over here, you should have this interaction between these 2 panels that can be helpful if you're trying to navigate a binding site and trying to orientate yourself. So I think I'm going to spend a little more time just talking through some of the features of the PDBe, because there's more things to discover that might help you in whatever you're planning to do. So we had discussed sort of 2 aspects of trying to find a good structure, a structure you might want to do docking with. But another aspect is the geometry, which can be evaluated for the model, and also the validation reports, which have sliders. And so another aspect of Molstar, if you still got your Molstar or PDBe, your web browser open, you can go to this instance. You just have to type pdbe.org and then /10j6. And this particular instance of Molstar, when we turn on, there's this option of the annotations to turn on the visualization of the validation report. And if you mouse over regions, you essentially get whatever the validation issue is. So if it's green, it means there was no validation issue. If it's yellow, it means there was 1 validation issue. And if it's orange, it means there were 2. And so this particular protein is neuroglobin, so it's essentially hemoglobin that's been optimized to hold more oxygen so that your brain, essentially it's hemoglobin for the brain. And this particular structure is a little bit of an older one, so it has a couple of validation issues. And this one in particular, Ramachandran outliers, just in case you've never heard about that before. So Ramachandran was a very clever person who was able to predict, even before we had very many protein structures, that there would be regions where this the bond described by these two angles here would only have certain allowable conformations. And it essentially has to do with the R groups hanging off the peptide and where they can clash and where they can't clash. And also like the backbone. So the clashing of this R group with this group here. And so there's certain allowable rotations, and regions actually also correspond to secondary structure. So obviously you can read and learn more about that, but in terms of if you see a random trend outlier or there's something to a bound length or a clash, those are regions where perhaps the model isn't as good because it's not meeting our understanding of how things should exist in 3 dimensions of a protein structure. So regions to just be aware of, so there's a bunch of validation flags near the binding site, and there's another structure out there that has the same binding site without those validation flags, and that's probably the one you want to go for if you're going to do some docking with the structure. And just to highlight that geometry and fit to the data, so again, talking about how data fits into a model. So here we've got regions which have random chambered outliers, and you can see they also are regions where there's not really good fit to the blue, that's the blue density map there in the structure. And just to quickly go through the validation slider. So the central square in green, these are all scores that relate to essentially the quality of the model, and they're applicable to any structure. So it doesn't matter which experimental method it was, these metrics will be there and are applicable. These metrics here, R3 and RZ outliers, these scores are actually particularly relevant to the method X-ray crystallography, and so you'll only see those sliders for X-ray crystallography structures. And so R3 is a measure of how well does it. You took the model and you back predicted, sort of predicted what the data should look like. How much does that data then map onto what the data really is? That's what R3 is in a nutshell. And then RZ is essentially a measure of how much the atoms are sitting within the electron density. And just to, again, remind you that you can see this when you visit structures. So, to change gear a little bit, so we're going to dig into the nitty-gritty of the structures, but now we're going to start moving up a bit and thinking about how do we consider all of these structures in context, given that we're now at 200,000 structures, which is a lot of structures. And so we have about 60K human structures, but actually, if you look at them, only 15K of them are different, excuse me. There's only 4K different families. So, and why is this happening? Well, essentially, people will solve the same structure using a different method. They'll solve the same structure with a different compound present. There might be different people involved. There might be different conditions, different pHs, different cofactors, what have you. Different mutations in the structure. And so, this is a structure I've shared before. It's essentially the structure that helps us digest and process chocolate. So, it finds some of the fatty acids that are present in chocolate. And we actually have 242 structures of this protein, and we have 227 different small molecules down because it was considered an interesting drug target at some point. And so a lot of medicinal chemists tried to develop inhibitors to target this particular small molecule. Unfortunately, no clinical trial or drugs that went to clinical trial came out of this structure-based drug design, but it is a protein that's had a lot of structure-based drug design done about it. And so in order to help navigate this sort of, rather than just visiting an individual entry page and look at each of those 242 structures individually, we have the PDBe-KB pages. So, this is a link, where's the link? Oh, I think I've taken it off my pages. There's a couple of ways you can get where you want to go, as it always is in the PDB. So often what I'll do, if I know the protein name, is I'll start typing the protein name or the chemical of interest. I think I said, yeah, binding protein 4, here it is. Oh, maybe the last release had a few more structures. I think the numbers might be a little off. Anyway, so if I click on this, we have the PDBe interface, and you can see there's, 267 structures, and I could click and view each of them individually. But another way to do that it's this one type of protein from one organism. So that means it has one of these identifiers and one page, which is the PDBe knowledge base page. And if you click on that page, you now have a page that, using the power of our APIs, is powered extremely by some wonderful analysis and aggregation. You now have an interface where you can look at all of these structures in context with each other. And so I really wanted to share this with you because I think if you're doing structure-based drug design and there's a cohort of structures out there on the PDB, you want to go visit this page and look at what they look like in context with each other, not just look at the individual entry pages or download them. And so some fantastic things that we brought online last year, one of them was these 3D view of superposed structures. And essentially we've done an analysis. We've looked at all 267 some structures. We've looked at how they relate to each other, and we've clustered them in terms of, so if you want to look at the diagram here, we've done some sort of analysis, and we've clustered them into two clusters after that analysis of slightly different structures. So you can see essentially this loop is subtly different between these two structures, and only one structure actually classes differently. So I think it was 267 structures. One is different, the other 266 are the same. You probably don't want to pick the 267th one to be the one you jump into. You probably want to pick the ones that are similar because this is the structure that this protein most often binds. Most often, it's a conformer, the protein most often appears to adopt. And so this is under the structure panel, but you can also look at it from a ligand point of view. And essentially what you can get is an overview of all of the small molecules in those 267 structures. Without even having to download the structures, you can view them all at once. And so I picked this protein because it's quite a simple protein, and it essentially binds fatty acids and transport ligaments. Had a lot of weird came down to it with the idea that it might be a useful target for heart disease. And so essentially we're all binding in the pocket where the fatty acid would bind. They have competitive inhibitors binding there. And it will take a while to load. Should probably have this loaded beforehand. So just again, so this is, you type it in here. You go to macromolecule, you see a lot of these instances. You actually also see it on entry page. So if you go to an entry page and you go down to here, in this section of structural analysis, there's a little PDBe-KB icon. Click there and it takes you to this aggregated view. And we have APIs underneath this that you could potentially call to if you're interested in a certain aspect. So it's actually a page completely devoted to the ligands down there, and you can click on, you can show all of the ligands in these little panels, so you can see all of the chemicals sort of by scrolling through, and you can see a set that is similar and that interests you. And if you want to look on the monster instance, you can just click on that panel, and if there's more than one structure with that small molecule bound, you can move between them, viewing how the experimental data supports that small molecule being bound in that structure. So it's really powerful. And another aspect to bear in mind, again, 267 structures, is that you have this as analysis of the ligand binding site. So that analysis window that you were interacting with in Molstar is also keying on this analysis. And so you can use and sort of zoom into which particular amino acids are involved in binding. And if you, it's here, you can get a sense of, so essentially I've shrunk them all down to see which regions are most often involved in this binding site. So if there is a cohort of data, then this is potentially a really powerful way of viewing it. And so there's multiple other options here to explore. One other thing I wanted to show you sometimes it's easier if you remember the gene name. So this is actually a project that I was involved in a number of years ago, trying to do structure-based drug design against this particular enzyme that is involved in to virulence and tuberculosis. And the reason I wanted to bring up this particular structure is this was a structure where docking didn't work. So this is a monomeric structure. So it is essentially, here we can see it's composed of a single subunit. On the entry pages, you have all sorts of ways in which you can look at that. And this is a part that we can, and I should say sometimes the thing to be aware of is in some of our pages will show what is grouped in structure, not what is the assembly. And so just be aware of that and look out for where we say it's a monomer. So that's kind of quite important to notice. And this is just an aspect of X-ray crystallography. Sometimes we'll solve more than one instance of a monomer in a crystal structure, and what we display is essentially what is the experimental data, not necessarily what's the assembly. Before I go any further, that actually also means that we have here, we'll have the files that correspond to what is in the experiment, and then we have these additional files which you can download, where we've actually just said, we're only going to give you what is the biologically relevant assembly. And because in this case there were two instances of that same assembly, there's two assembly files that you can download. Just a little aside to help navigate our pages. So where was I? Okay. Here, right. So if I look at these 14 structures here, sorry, I want the 3D view. One of the aspects you can see here is that essentially this dashed line means that there was regions that in certain structures you couldn't model. So I was someone doing enzyme assays on this project, and someone else was doing the structural biology. And if I turn on all of these instances, and we see all of the structures, then I can actually export all of those models into a zip. Go to my downloads folder, grab that zip, and just drop it into the monster instance. Boom, booyah. And then essentially, I can load all of those structures in one go. And so that's one way in which you can sort of leverage the fact that we've got this aggregated knowledge. I should also mention that you can load AlphaFold structures in here. So if you've only got a structure of a certain domain, and AlphaFold has structures of other domains, or regions that just for whatever reason aren't present in the structure, sometimes it can be interesting to see what has come from the AlphaFold database. In this particular context, as we were saying, we did docking, and we did medicinal chemistry based on sort of what we had done with the analysis of the binding pocket and the docking, and it turns out essentially the protein changed its shape. So as we solved the structures of all of the chemicals that we developed based on our analysis, the protein had changed its shape. And one way that I can show that is I'll turn off cartoon representation, and I'll turn on a representation called Putty, which shows B factors. And if we zoom into where, here it is. Grab a different one. There's one of the ligands. So essentially, you see where all the ligands are bound in this pocket, quite adjacent to it is this massive uncertainty represented by these snakes that have got really fat, or they've simply disappeared because we can model the structures. So essentially, this particular protein, half of the binding site, or one of the walls of the binding site, essentially disappeared. And as we tried to grow an R group into a hydrophobic pocket, the whole thing flipped around, and R group went out into space, and the chemicals weren't any more potent. In fact, they were less potent because this protein had changed its shape and was actually in a less energetically favorable shape in order to accommodate this different binding of the set of compounds that were developed. So let's just go back here. So this is this instance here. There's the small molecules bound, and as you can see, there's this region of the equation that sort of just disappeared / moved out of the way. And so what we were trying to target kind of disappeared. Now this is the fatty acid binding protein. And as you can see, this particular, even though there's 267 structures, there was not very much change. Whereas with this particular protein, there was only 14 structures and there was dramatic change. So this is another reason why it might be worth visiting one of these aggregated pages if your target has experimental data, because it could be that there's going to be conformational changes and you want to be targeting the right conformation, or you want to be aware that that conformational change might mean that your design strategy isn't potentially going to be as effectual as it could be. In this PowerPoint, which I believe you'll have access to, I've also got a link to a YouTube video going through some of the details related to the calculations you do to generate the superpositions. I think this is a fantastic thing that is just available to you instantly online. It's pre-calculated, you can go and see and instantly get some context to not just one structure, but multiple structures compared to each other. And I think that is me covered, and I believe I've almost slightly gone into your coffee break by maybe 5 minutes. So just to finish by saying, one of the things about these KB knowledge pages is we have essentially used our APIs in our analysis to pre-cluster and pre-calculate things that we think might be interesting to you about multiple structures. So please take advantage of them and utilize them in whichever way is beneficial to you. And I also wanted to just thank, this is just some of the members of our team, of software developers, and people like me who work with the data that's being deposited. And I just want to thank all of them for the teamwork that we do together to continue to provide the PDBe. And thank you so much for listening. Any questions, any feedback? And while I'm saying that, I'm just going to quickly put up my, well, if you can hold on a little bit for, where is it? I would love to get some feedback. So I have a feedback slide. We'll get it to work. And if you just have a quick moment, I think there's just one, maybe 2 questions about the course so that I can get feedback. Get it to load.
As there are some final questions in the audience.
And maybe some final questions while people take the time to visit. Yeah.
It seems there are no questions. Everybody is satisfied. This is a great lecture and tutorial. So thank you very much.
Yeah, thank you.
In silico drug design, it is a challenge. So best of luck.
Thank you. So we can make a break for 15 minutes for coffee, some refreshments. So we can find it on the last row of tables.
Tady vám je tady teda a jenom po naší povídce. To je jenom zavřít, mám už tady. Toto, prostě tímhle dát, vytáhnout ze zásuvky. Toto je rovnou. Jo? Pánové, v kulize začnu teda.
We'll start in 5 minutes.
Abych vás jako nehodil, mě to je
To je skvělý. Si udělám to kafe. Chceš taky něco chytí?
Goes to the left?
Yeah, to the left and then...
Tady
Center of excursion.
I'm not pretty sure that because they have some resources.
What are you studying? I studied technology. And I also can study.
Oh, yeah. Yes. My very close friend from Yeah, exactly. So that's why I know. And he also studies in because he's the first year wait, now physics and then yeah, and you know, like we've been to Olomouc because he just wanted to like, oh, it's such a good university, we have to go, and then we went and we were so far away. It's like you go into nowhere and like at some point there were no buses and we were like, okay, but it was so great. I was living in Barcelona
So, 5 minutes has passed. So we will continue with the program, not to make our delay even bigger, because we still have 2 lectures today and then a poster session as well. So let me introduce you. Marian Novotný from Karel University, who is my colleague from Elixir Czech, and he will speak about how to find binding sites and how to make some art on it.
Okay. Thank you, Karel. Thank you for inviting me. Good afternoon. So I will try to speak about binding site identifications.
A little bit louder.
A bit louder as well. Sorry, I was thinking. I was telling my colleague today morning what I'm going to say, and he said that he definitely has to look at the video because he doesn't believe I can do it in the 45 minutes I have. So I might cut few parts, and okay, this should work though. Should it? It should, but it doesn't. Where's the trick? It's frozen. Yes, thank you. So, if we want to talk about binding site identification or prediction, we have to probably define what it is. You intuitively will understand what binding site is, but let's say that it's a macromolecule. It's a place in a macromolecule that interacts. You can talk about forming contacts with other molecules, and it could be, if we talk about protein, it can be other protein, but today we will focus on small molecules, which we would call ligands. And why is this interesting? Obviously, this is a drug design course, so structure-based drug discovery often starts with identification of suitable binding sites, but it can be also functional notation of variant effect prediction and many other areas where this could be used. But if we will today talk about structure-based discovery, and the binding site typically has some properties which might be or might not be easily used in the prediction. And you can do it on the structure level, or you can do it at the sequence level. And nowadays, it's so easy to do it on the structural level, so we will again focus on the structural level. And there are very many approaches to this task. You can simply try to do it manually, even, and you can do it on two levels. Manually, you can look at the multiple sequence alignment for the protein you are interested in, and there's a good chance that the parts which are the best conserved will also be the binding sites. Or you can look at the structure, and the old knowledge, it's really for many years known that if you look at the enzyme structure, and you will find the biggest pocket on its surface. And again, we can discuss what finding biggest pocket mean, but typically when you will have a structure, you will be able to do it on your own. That would be probably the place where the ligands bind. But you can have also more sophisticated and crucially automated approaches. And these, again, would fall into various categories. They would be template-based or template-free, and the template-free would be either geometrical or energy-based or statistical or machine learning, which in some way would combine all of those previous ones. And there are over 50 methods developed, and the comparison of those is extremely difficult, so it's not basically done. So I will be telling you about one specific tool, our tool, claiming that this one is the best. But you don't have to believe me, but it will be very difficult for you to prove me wrong. And I will also show you why. So the approaches, there are two standard approaches. One is that you really try to locate cavities, so it's a geometrical approach, or you have some sort of probe, and with that probe you try to touch various parts of the protein and measure the energy and find those that produce favorable interactions. And then there are machine learning approaches, and then I will want to talk about one of those, which is called P2Rank, and that's a method I will be talking about, but I'm probably not the best person. The best people to talk about it are my colleagues from Mathematical Physical faculty, Rado Krivák and David Hoxa, who have developed it, and I joined them only later in this endeavor. So I will try as well as I can. So what this is an algorithm which takes a protein structure as an input, so it's structure based, and built a supervised machine learning model which would predict the binding sites on the surface and visualize them on the surface. This is already quite important distinction. Many of the programs which are machine learning concentrate on not on the binding site but on binding residues. And so it defines the set of binding residues. It's a classifier which says this residue is part of the binding site, while we try to predict a binding site. And you will see the distinctions in a while. Okay, so what we did, we obviously started with some data set which we used for training. This was a data set of known protein ligand complexes. I will get into details a little bit later, but it's important to say at this point that for us the ligand has a specific meaning. It comes from the data set which we used. It was a Chen data set, and that does include small molecules, but not too small. So it has to have at least 10 non-carbon atoms. So in fact, a typical ligand for biologists, as myself, would be a metal ion. But this is too small for us. So we cannot predict metal ion binding sites, but we cannot also predict the bigger things like pieces of nucleic acids or a peptide, right? So that would be like typical drug-like molecules would fit into this, but we can't predict two small things or two big things either. So what we do when I said we do the prediction of binding site, we take a protein represented as surface accessible area, and then we put a mesh on it. And the mesh has regular spacing with points. I think it's something like this. The spacing of the points is an attribute which can be modified, but the point is that for each point here, this collects information about a neighborhood, which is 6 angstrom, so quite a big neighborhood. And for each of these points, based on the training data set, we can label the points as binding or non-binding. So these points are not part of the protein. These points lay on the surface of the protein, and they might be at the same place as some atom of the protein, but more typically than not, they won't be, right? But the idea is that it's a regular spacing distributed on the surface of the protein. And for each of these points, you would extract vector of physical, chemical, and structural features, and you collect it, as I said, for this area of 6 angstrom. And then, based on these properties, you would build a model. And this model then is able to classify if this point is or is not part of the binding site. And so what features do we have? We have about 30 features. Most of them are very simple things like, is it hydrophobic, is it polar, is it hydrogen bond donor, is it hydrogen bond acceptor? And you collect it, for each point you collect it for this area of 6 angstrom and do some average value for this. And I think I have a list here. So of the features we collect. So you can see here these are basic properties. And then there is even, for X-ray structures, we collect B factor, which turns out to be a complicating factor when you get to alpha-fold structures, because we can do these predictions also on predicted structures like AlphaFold, but they do not have B factor, right? And even more in the B factor column, they collect the information about reliability of the prediction. So when we started predicting on AlphaFold structures, we had to give up this parameter, so we had to build a new model without it. And then there are structural properties, and one of them is called protrusion, and I will talk about that one, because this one seems to be the best we have in our model. And then we also can collect information about sequence conservation. So you can either run the model just based on these 30 features, or you can add sequence conservation. And we started with a very simple pipeline to assess the conservation now, but it was extremely slow. So now we have a new one based on Hammer, developed by David Jakubec, who did that at IOCB. And this one is significantly faster. And again, in a while, I'll show you what it can do and how much faster this is. And we have all this, and then we can use the model to predict the binding sites on any available structure. And as I said, it can be experimentally solved structure, but it can also be predicted structure. And you do the same thing as I already described. You take the structure, cover it with a mesh of surface accessible area points, and then you apply the model to every point on the mesh, which would give you something we call ligandability score. And then you filter out all the points which have low ligandability score and cluster those that have high enough score to form a binding pocket. So we predict binding pockets by finding close enough points with high ligandability score. And then you can score the pockets and do some other filtering, but in the end, you would map those that are promising on the surface. And it would look something like this. So you see that we have, in this place, we have green points and red points, and the red points are those which have high enough ligandability score, and that were clustered into some sort of binding site. And then we map on the surface of protein by finding residues on the surface or in the protein which are close enough to these points. And close enough for us is 4 angstrom away or less. Okay? So this would be output from our program. But you can do all sorts of predictions, but the question is whether they are relevant and how to find out. And here you get to, I think, murky waters. So if you would say that you do the predictions, as we say, for the binding site, not for the binding residues, it is not really easy to use confusion matrix because there would be so many different surfaces you can define as non-binding. It's not that they would be infinite numbers, but it would be really high numbers. So we cannot simply use it for the binding site prediction. So we use something different. I will try to explain now. So we use something like a success rate, which works in respect to how many binding sites you have in the structure. So if you would have one binding site, we would look into either first pocket that was identified or what we prefer to give it some room for error. So we would say n plus k, and the k is our room for error. That means how much time you have to investigate your results. We would typically do n plus 2, and you will see that in our results. So if there is one binding pocket, we would look in the first 3 predicted by P2 rank and check whether these are in alignment with the known binding sites. Okay, so that's how we do that. Yeah, so this is once more, we call this parameter DCA, distance from C alpha atom, and that's the distance is between the center of the predicted pocket and any atom in the ligand. And yeah, again, this top 1, top 3 is something which we would often use. But if you would have 5, of course, binding sites, then this would be, we would look into first 7 in this case. Okay, and then I have to talk a little bit about how we did the training. You would, when you have machine learning approaches, you would typically split your training data sets into 2 or 3 data sets, depending on how sophisticated you are. And my colleagues are from mathematical physical faculty, so they are extremely sophisticated. And that's why they use 3 data sets. One is used for training, one is used for validation, and validation is meant to improve the hyperparameters in the machine learning, like how many layers you will have in your neural network, or how many, in our case, we don't use neural networks, but random forest, how many forests you would use to, how many you would use to have it random enough. And then we have a test set, which should not be used in any part of the training, but it's part of the data where we still know the results, where we still know what is the true binding site, and we can look and check how good our machine learning approach is. And to do that, we actually had a number of data sets. The data sets for machine learning in biology, I think, are the real bottleneck for anything. You would not have enough data, you would not have high quality enough data, you would have all sorts of errors inside, and then you end up with things like this. So we used a training data set which was called Gen 11. I do have links to all these data sets, which in the end was a data set which was supposed to be highly curated. It's from 2011, so by the time we had maybe 150,000 structures, maybe over 100,000, but we would still get only 251 proteins with ligands which are good enough to go into the data set. So you see that almost all, like 70% of proteins in the PDB contain some ligand. So there should be something like 70,000 structures with a ligand. They did some sequence filtering, so they didn't want to have one protein 50 times in there, but still you end up with only 255 proteins. And that was our training data set. Then we had a validation data set with few more structures, which were different data sets collected along the way by various scientists who deserve a lot of credit for this. And then we had a testing data set, which were two more testing data sets, Coach and Holo4K. This one was composed for authors of one of the methods for binding site prediction. It's called Coach. And then there are another one, by far the largest. Holo4K had about 4,000 structures. So you see that actually our training set was relatively small to the testing set we in the end had. Okay, and here is a comparison with methods we thought at that time were state-of-the-art. One of the most typical ones is called FPocket. It's one of those geometrical models who try to fit something they call alpha spheres to identify the pockets on the surface of the protein. And then there's one called Sidehound, which is one of these energy-based, which puts probes and taps on the surface with various probes finding favorable energetic interactions. And there are always something deep side, and deep pocket, and meta pocket. And then we use our DCA statistics to describe in how many % of the structures in this 4,000, in case of Holo4K, we were able to find the binding pocket among the top n or top n plus 2, right? And this F pocket plus prank was the original version of our algorithm, which was just not able to predict, but was able to score the predicted results from F-pocket a little bit better than F-pocket itself, as you can see here. So you see that's a % of success. And we can show here that P2 rank from these programs was the best, at least at the time when it was published. So we could do like 72% of the binding sites that were predicted were for 72% of these 4,000 proteins, in 68% of these 4,000 proteins, we could predict the best binding pocket as our best binding pocket. And if you go top n plus 2, it improves a little bit, but you see that not substantially. I would say it's enough to just work with one predicted pocket, or if they are free, then with free best. And people often talk about interpretable AI, about interpretable machine learning. So we also wanted to know which of these 30 parameters we use have the strongest signal, strongest discrimination signal, and it is the protrusion. And protrusion is very simple geometrical parameter which asks how many other atoms you can see within, I think, 6 angstrom. So the idea is the less you can see, the more protruded this residue is. And that simply measures how much space there is around each atom. And this was single most useful parameter we had in our data sets, and there are many other things we use, but they are nowhere near as important as protrusion. And then we were also interested in whether the conservation is any good, whether by adding the information about sequence conservation we can improve our model. And again, you would see here results for various methods, and then we just add this line here. And you would see that it has improved, at least in the top N, a little bit. Again, actually quite a lot in terms of how much we had before. But what we also see is that it helps us to, it doesn't find many new binding sites, but it helps us to rank them much better. So when we use the conservation, you will get much better ranking of which binding sites are really the important one in the protein. We still struggle a little bit, and that's the other picture, with number of predicted binding sites. Because if you want to have a method which would find very many binding sites, then the best option is just to predict many binding sites. So you would have a lot of false positives. And that's actually something which you see on many of these methods. So this is the average number of binding sites predicted by various tools. And you see that the F-pocket, for example, predicts 15 binding sites, which might or might not be very useful. And then the other methods predict less and less. And you see that the P2 rank still predicts typically 6 binding sites, which is high number. But if you use the conservation, it drops to a little bit over 3. So the conservation doesn't find new binding sites, but allows to score them better and to little bit reduce their number, which is probably worth. Okay, and then this is just another graph which shows how fast individual programs run, and you could see that some of them are actually quite slow, like Coach, and P2 rank is in second, so it's one of the fastest, if not the fastest program there is. So it can do the prediction really fast. On the other hand, from my experience, whether something takes 6 seconds or 3 minutes is not really an issue, right? If you are really, if this is supposed to be the drug which changes your life, you can probably wait a few more minutes to get the good results. So I don't think it's extremely relevant. It just shows that these random forest machine learning approaches can be extremely efficient and extremely fast. Okay, so how can you use the program? You can of course download it, but there is also a web server run with the support of Elixir. I'm supposed to say that, I guess, Karel, to promote Elixir as much as I can. So it runs on the web server, and I will see how fast I am. I was thinking that I will show you how this works. I hope I still will have time to do that. Okay, but I want to talk about one more thing which wasn't announced, but I hope you will find it useful when you think about drug design and binding of proteins, and that is to which structure you should try to dock. You can identify binding site on each structure, but for many proteins you will have a number of structures available. And then you will get this burning question which one you should use, whether all of them or just selected ones, and how you can do that. So we came to this working on Prank Web, and the idea was that the colleague who is developing the program, Rado, was always complaining that the program is too good, that it shouldn't be so good, and that we probably do something wrong. And he said maybe we are not testing our tool on realistic enough cases. So what we typically would do, and that's all the structures in the holo or Chen in other data sets, are the structures of proteins which already bind a ligand. So there is never an apo structure which is not binding anything. While if you would want to predict function of a protein, for example, you would, if you already had the ligand there is nothing to predict, but you might want to predict which ligands would bind, and then you would probably do it into apo structure. So we were wondering whether we shouldn't test it on the apo structures. We said, okay, why not? We can do that, but we found out that we can't, because it's not really easy to get the list of apo structures for a given list of holo structures. We first thought it would really, we would just go on internet, find the tool, download the list, run the program. But it turned out that it doesn't exist. There used to be such tools, 2 or 3, but they all disappeared. And again, coming to how long-lasting are these tools on internet, typically not very much. And I don't know why these tools disappeared. I might find out, there might be some cares, but so far we are still holding our tool. And so we had to do that, we had to develop our own tool to do it. So we do have now a tool which can, for a given binding site, and I think that's really crucial, not for a structure, not for a protein, but for a given binding site, it would find you all the other structures there are that contain this binding site, but without a ligand. Or simply all the structures that contain this binding site and classify them according to whether they do or do not contain a ligand. And so, yeah, so that's the tool which we have and call Ahoy. And why are we interested? So this is example which might be, again, interesting for the drug design course, and these are cryptic binding sites. These are the binding sites where we think our tool should fail, because in the holo structure there is a nice pocket, like in this case that's a M38 kinase, which has a nice lipid binding site, which you can see in the holo structure. But if you solve the apo structure, it looks like this. There is no pocket whatsoever. It just doesn't exist on the apo structure, and it should not work. And it doesn't. I will show you. If you would run P2 rank on this structure, you will not find this binding site. If you will run it on the holo structure, you will find the binding site. Again, I can show you that. But there are other cryptic binding sites. So the idea is that there is a substantial conformational change, which is either induced by the ligand binding or has to happen before the ligand binds for the ligand to bind, right? There's another example with the nuclear receptor which binds heme, and it is a bit more subtle here, but still there is a change in the orientation of this. This is helix in this structure. It's a bit more disordered without the ligand. And the conformational change doesn't have to be big. It can be just one side chain changing its orientation. So either it's like this and it blocks the entry to the pocket, or it's like this and it's open, right? So identification, these things can help to, for example, repurpose drugs. If you find new binding sites for the drugs which are already existing, it might be helpful. So just to briefly show you how it works, it's very simple. For whatever binding site you can think of, you can define it by PDB code, chain, and the ligand name. And here is the interesting thing, maybe for biologists among you, it doesn't have to be a ligand. It can be anything which can be described by this simple code. For example, here it is phosphotyrosine. So this thing doesn't have to be ligand. It can be post-translational modification. And you can find all the pairs of the protein which are or are not post-translationally phosphorylated on specific site. And you can, this is just type, but you can also say specific position. If the structure would bind 3 hemes, you could say which heme binding site are you interested in. And then it will start the search. It will do something like this. It will find all the structures which don't bind, which don't have phosphotyrosine here, all the structures which have phosphotyrosine. And I would like to stress again, how is this different from if you would go to PDBe and you would ask for structures that have this? The difference is that for the Src kinase, as this is a Src kinase, you would have about 600 structures which contain just SH2 domain or SH3 domain or kinase domain, but do not have this site. So they would be apo, but are not useful for you because they are not showing the binding site. But here you look specifically for the binding site, and it only shows you structures which have the binding site conserved to a certain degree. You can define how much conserved it has to be. You can say it has to be fully conserved. You can say from 80% and so on. But it shows you whether the binding site is there, because there are so many structures which, for example, contain just this bit, SH3 domain, but the binding site is simply not there, and these are not useful for you. And you can, of course, download the results to something like PyMol and look at them in more detail. So this previous slide, it's more just to give you an idea. You can toggle on or off the structures, but it's light mol. You have tried to work with it. You've seen that it's not really easy. I don't know anyone who likes it, but everyone is using it because it's so bloody fast. But if you really want to analyze the structures, you can download them and use them in, for example, in PyMol. And so we thought we would go a step further and we would try to build a database from this. And that would be a database of precalculated apo-holo pairs for individual binding sites for biologically relevant ligands, where you could be able to search not only by the binding site, but also by UniProt ID. So you could say, show me all the binding sites for for hemoglobin, for example. Or you could say, search by ligand ID. So you could say, show me all the apo-holo pairs for hemoglobin, right? And so what is biologically relevant ligands? That's an, I think, important question. It's not easy to say, but here we rely on another database, which is called BioLib, as the database of biologically relevant ligands developed by this Heng group. And they say that these are ligands which I think the most crucial feature, there is a list of things which they don't consider biologically relevant, so it's a blacklist. That would be typically the things which are in crystallization solutions to help proteins to crystallize. And these are on the blacklist, so these are not shown. And then they say that for the ligand to be relevant, it has to have more than one contact with the protein. So there are all sorts of weird things in the PDB where something hangs by thread through one contact, and these again are filtered out. So there would be about 500,000 such biologically relevant contacts in the PDB at the moment. And this is the interface, again, very simple. You just can search either by like in Ahoy, or by UniProt ID, or by ligand identification, which I tried just penicillin in this case. And then it would, sorry, it doesn't go in steps, but first it gives you a list of all the binding sites in different proteins, in different structures which have penicillin. And it always shows you how many apo and how many holo structures there are. And I think that's already useful because you will find a lot of structures where there is no apo structure and cannot be simply used for building data sets of apo structures, which I think is the main idea. And then you can, of course, go to view, and then you will get this view again with a detailed list of structures, either apo or holo. But you can also look a bit more in what we have. And so this is the list of the most common binding sites there are in PDB, and you would see that they are mostly occupied by ions, metal ions, so zinc, magnesium, calcium. But there are also huge things like bacterial chlorophyll, heme. And this is interesting, I think. This is the percent of entries which have apo structure available. And you see that it differs quite a lot. It's never very high. I think the highest is like 70%. But here, for example, we have a lot of structure of bacterial chlorophyll. But we have no structure of this photosystem without bacterial chlorophyll. And indeed, a lot of people who are interested in cryptic binding sites, they would say that the very strong indication that a binding site is cryptic is that there is no apo structure available. It's simply too difficult to solve the structure without the ligand. So that could be indications of these cryptic binding sites as well. And so just to conclude this part, I would say depending on what you want to do, it might be a good idea to test your tool not only on the holo structures, but also on something more difficult, which would most of the time be the apo structures of the same proteins. And we believe now we have a very specific tool which can do it for the ligands, for post-translational modifications, even for the water molecules. I didn't tell you that, but even the water molecules are well described in the structure, and they are often extremely important for the stability of the ligand binding, for example. So you can ask, is this water molecule present in the structure? Is it conserved? And then we are building a database. We should actually send the manuscript tomorrow or on Wednesday if everything goes well. So there should be a preprint on Wednesday available. And I'm already running over, right? But I was thinking I will show you at least how the prank web, because we have a new version. We were working on it to deploy it. But now it will not be. I think I need to share the screen again. So the prank web has very simple interface, and I thought I will show you an example with the. So the structure I show you is SAR kinase 2 SRC, and you can either use the conservation or not. This always makes it a bit longer. We now have only the pipeline I have described, which is using Hammer. Before we had an option for the user to upload the conservation scores as they like them, but no one was using it. And it was too difficult to keep it, so we just threw it away. Sorry. But I just, for the sake of the time, I hope whenever you do demo, nothing works. So let's see what will happen today. So this is the, again, it's small star. So it shows that's the ranking of the pockets. You see that the red pocket is so much better than all the others. And indeed, there is a, of course, when you do the prediction, all the ligands are thrown away, but then they are shown again for the visualization. And this is the ligand, ANP, which is analog of ATP. So, and you can zoom and all sorts of things, but the new version now is that we allow to do some simple task on the pockets. So you would, assuming that there's an apo structure, you could try to dock something in there. So we have two tasks which you can do. First is measure volume. You can do that, that's very quick. But you can also try to dock in there something. So you would and it doesn't make sense to dock in the structure which is already binding. But you have to use the smiles and create task, and it will use autodock Vina, very simple docking protocol to dock something in there, so you can have a look and again download. And I will go back because I have used Ahoy, and I know that the APO structure for this is one FMK. Yeah, but
may I have a question?
Yes.
If I use the command line interface version that runs on a computer, does it also have a backend for Vina, or I have to use Vina separately?
That's a good question. I think you will have to use it separately. But now you could do that, except could you create task, you would go to MSDChem. That's what I would do. There is probably a better way to do it. I would go to MSDChem, not tutorial. Sorry. Ah, yeah. I know what's the problem. My bad. A and P. Search. Copy this, which is smiles description of the ligand, and go back, call it whatever, enter here. Okay, and then you would create task. I will tell you that this is not going to work now. We were a little bit rushing to put it on web to show you, and it turned out that it's queued and it will stay queued forever. There is some bug in our program, so it was good we found out. But it works if you don't do it this way. But if you would, there is something in the PDB file which is wrong. But if you would upload MMC file through here, sorry for that, we will make it running. But if you would upload MMC file here for FMK, it would be running. But I can also show you real quick the difference between the APO and holo structure. Let me check 2NPV, I think, is the APO and holo structure for the cryptic binding side. Yes, 2NP. Submit. And the best binding pocket is the one up here, which is the one which is binding the lipid. Okay, you see it's clearly better. If we go to the APO structure of this, which is the code, let me check, 2ZB1. You will see that in this region there is no pocket now, and the best pocket is binding some analog of ATP because it's a kinase after all. But this pocket is simply not present. Having said that, we've tested Prank Web on a data set of cryptic binding sites, and it can, even in the APO structure, predict like half of those. But there are definitely cases where it can't find them, right? And this is one of them. It's simply, it just doesn't predict anything in this region. It's simply probably the tunnel is not there, there is no protrusion which could be measured. So that's why it probably doesn't work. And that was very brief introduction of our tool. So thank you very much for your attention.
Are there some questions?
Yeah.
Oh, hi. Sorry. It's really nice work. I mean, I really like your talk a lot, especially I like the last part about the alpha structures. I was just wondering, because obviously machine learning, like I think there is a lot of, I think there's a lot of like bias against the predicted structures, and I was wondering if you plan to do any collaboration with a structural biologist to validate this alpha hollow structures, if they can actually fit the ligands.
If they can fit the ligand.
Yeah.
So what I think we are a little bit limited, but what we can do is that we can try to compare whether there is a difference between apo and hollow structures. Then it's a bit complicated. And then there is the whole field of the AlphaFold structures, which are not well annotated. So in terms of apo-hollow, you don't really know what you get. You might get apo structure, you might get hollow structure. There are some first indications that most of the structures you will get from AlphaFold are indeed hollow structures, because that's what's like 70% of the PDB is of proteins with ligands. So it tends to learn from what it got. And we now have the collaboration with the team of Thorsten Schwede from Basel, who is trying to do the analysis of ligand binding on AlphaFold structures, and they simply ask us to generate for them these apo-hollow pairs where they could test it. So we see the idea that it could help to build very specific data sets which can help to clear things out. But we are not as much in the position to give the answers ourselves, I think.
Thanks.
Other questions? I have a question regarding this tool, and do you think to combine this and these simulations to analyze trajectories, maybe this will give you more information?
You mean the cryptic binding sites? Yeah, well, we are thinking about it. We are not big experts in this. We do have now a postdoc who is good in this, but it's difficult to do it on scale. That's the problem. If we would want to run it on PDB level, then you would have to do a lot of simulations. And we did try to do it properly on number of structures, and we still don't have long enough simulation to see the transition from apo to hollow in most of the structures we have. So it seems to us, in our hands, you would need much more computer power to see the difference. So we thought we will use it. We will try to use it still, but at the moment it looks like it's difficult.
My idea was that you can maybe guess using these machine learning methods even from the beginning of the trajectory that this side will open.
Yeah.
100 nanosecond, one microsecond.
We don't see that. We see just the opposite. That it looks like this sides. It looks like it will move somewhere else. That the binding site itself is relatively stable, but the flexible parts are on the brinks, on the sides, and this will eventually, like this, move. So what we see so far is that there is, of course, increased flexibility in the protein chains with the cryptic binding sites, but it's probably outside of the cryptic binding site, which is not helpful much in identifying the binding site itself, but it is definitely a way to go. But I think we need more experience and more power to do that.
Yeah. Thank you. Some other questions? No.
Yeah. Actually, on the user experience point of view, you're imagining to write also some easy Python API so that all these command line functions can be accessible from a function in Python or some sort of...
It's in the list. It still hasn't been done, but of course that would be, especially for the database, I think we need to do that simpler.
So that, like, for example, from a binding site, you could retrieve information about the box or like the amino acids, so that it's possible to dock on any kind of programs.
So it's already, you will get this information about binding site. If you download the results, you will get them, but not on scale, right? So we really need API to do it en masse. Thank you. Karel.
I have a question. Would it be useful if from those found active sites would be run let's say, channel? Calculated with mode?
Yeah, channel is always great. It can be used to almost anything, right? Yeah, it would be. Yeah, it's also, it might help to connect the apo and holo structure in a way. Yeah, maybe.
No other questions? So we can thanks Marian once more.
Thank you.
For the next speaker. From today are already there. This.
Super, super.
And
jo, super.
And now we will have the last lecture from today, and it will be from Alžběta Tuerková, and it will be about how the biotech startups world is different from the academic ones, and how computational chemists can shape something in there.
Yes, so we are a startup that is focused on proximity-inducing compounds, or also known as PROTECs. And we are based in Graz in Austria, where we also have chemistry and biology labs. And we have currently opened a new site in London with office and with business development. So I would like to introduce you to what we are doing in there. And this is the structure of my talk. So I would like to start with an introduction into targeted protein degradation, which is a new modality to target so far undruggable targets. And then I would like to move to explaining a bit more about ternary complex prediction, which we use as a proxy for designing novel proximity-inducing compounds. So I will talk about challenges connected to this approach. I'm going to talk about how we apply ternary complex predictions at different stages of our drug discovery pipelines. I will also outline a bit more into about the methodology. So we use more AI-based but also physics-based approaches. And of course, I'm going to talk about validation of the approach, which is the major challenge, major bottleneck of all the computational approaches. Then I'm going to talk about linker generation, where we basically have a model for systematic design of different linkers to account for multiple different properties, such as solubility, permeability, structural novelty, and so on. And then in the last part of my talk, I'm going to talk about how is it like to be a computational chemist in a biotech startup, and how is it different from academia. what are the skills that you learn in academia and you can transfer into industry. I'm also going to talk about a bit about how the job interview process looks like, and also about different roles as computational chemists in the industry. Okay, so let me start with the introduction. So, as I already said, and it has been said multiple times during today, most traditional small molecule drugs deactivate functions of proteins. This is how they act. And so far, most typical mechanism of action is inhibition. But the problem is that when we look at the human genome, we can see that only 20% of all proteins are inhibitable because they have very well-defined binding sites that can be targeted, while the rest, 80%, is so far undruggable. So we have to look for other modalities, other approaches to target these proteins. And one of these approaches is targeted protein degradation, which uses natural cellular mechanisms found in human cells that are able to remove dysfunctional or misfolded proteins. And one of such systems is called ubiquitin proteasome complex. And what we do is that we design degraders, which are basically the drugs that can take advantage of this cellular pathway by hijacking these systems in order to take specific proteins that are then subjected to degradation. So this is shown in a closer detail how the ubiquitin proteasome complex looks like. So when you look on this picture on the left-hand side, this is the entire ubiquitin proteasome complex, where we basically take the pathogenic protein, or in this talk I'm going to talk about it as a protein of interest, or POI, that needs to get close to an E3 ligase, which is the pink structure. And the E3 ligase in turn interacts with multiple different units in this complex, where E2 conjugating enzyme carries ubiquitin, which is the yellow structure. And the whole point is that by creating this ternary complex, the protein of interest can get ubiquitinated by ubiquitin transfer, and such a protein is then recognized by proteasome and is being degraded in the proteasomal pathway. So what we need to do is that we need to design drugs that induce this proximity, so that's why we call them proximity-inducing compounds. And they are composed of three major components or fragments. So there is a fragment that interacts with the E3 binding moiety. Then there is a fragment that interacts with the protein of interest, which we also called warhead. And of course, these two fragments have to be somehow connected. So we have a third part of the molecule, which we called linker. Okay, and as I already said, there are multiple advantages and benefits connected to this kind of modality. So when we compare other types of therapeutic modalities with degraders, we can see that degraders are stable. We can use them for so far undruggable targets, as I already said. They work directly on proteins. They can be clinically validated. They are orally bioavailable, and because they are highly specific, we expect that they have lower toxicity and they are working under lower dosages compared to other drugs. But on the flip side, there are multiple challenges because there are multiple factors that come into play and then that determine success of degradation. So if you look at this equation, what we see here is that we have to optimize the PIC compound for solubility, for plasma protein binding, permeability, ability to form a ternary complex and get ubiquitinated, and also it's dependent on the POI synthesis rate. So what we do at Celeris is that we can predict and model some of these factors computationally. So for example, when it comes to solubility and permeability, we have a linker generator model where we can generate a bunch of linkers based on user input properties, and then we combine it into a PIC compound. And in terms of assessing formation of a ternary complex, we do ternary complex predictions. And then if you would like to look at ubiquitination, we can model the entire complex and measure a distance between the solvent-exposed lysines on the protein of interest and the ubiquitination sites. Okay, so let me move to ternary complex prediction. So as I already said, there are multiple challenges connected to this approach, and one of them is that we have to account for all the key interactions that come into play. So most specifically, there are three major kinds of interactions. So there is a fragment protein interaction, which is the interaction between the individual ligands and the respective proteins. Then there is protein-protein interaction between the E3 ligase and the protein of interest that are often transient and very dynamic. And of course, the overall ternary complex is also dictated or determined by a given linker, meaning that the length of the linker can basically dictate how the ternary complex will look like in the end. So what is another challenge, or let's say key determinant of ternary complex formation is protein flexibility, which is a known problem in structural biology. So here just an example of a CRBN ligase showing that there are multiple structures that possess certain structural variety or diversity, especially at the regions where we expect that the protein would interact with the protein of interest. And then here is an example of a bound structure from PDB. So the idea or the challenge here is that we don't a priori know which of these conformation is the one that is relevant for adopting a ternary complex. So we have to somehow account it in the pipeline. And of course, the third challenge is about validation, because there is just a very few number of experimentally determined ternary complex structures. So far, there are about 22 structures in PDB. And another problem is that all of them come from X-ray measurements, which can pose certain challenges, such as, for example, that we only look at a single snapshot of a structure, but not on the entire ensemble of alternate conformers. There can be some crystal packing effects that can affect both protein-ligand and protein-protein interactions. Of course, we are facing also some limitations when it comes to resolution, because most of the structures for ternary complexes are resolved with the resolution of about 3.5 angstroms. And there can be some other experimental artifacts that are always linked to X-ray crystallography. So right now I would like to show you how we can use prediction of ternary complexes at different stages of drug discovery pipeline. And we start directly with target identification, where we can use TCP to see whether it's even feasible to adopt a ternary complex with a given E3 ligase for which we want to develop a PIC compound. But we can also go a step further, and we can look at multiple different POIs, and we can also look at which of the target would selectively bind or form a ternary complex with a given E3 ligase. So when it comes to hit identification, we can run a ternary complex prediction with an SAR series of different of molecules with different linker lengths. So we can determine what is the optimal or minimum linker length needed to adopt a ternary complex. And we can also look at differences in terms of different warheads or different binding pockets, different E3 ligases, which we can evaluate by running ternary complex predictions. And the same is true for hit-to-lead optimization stage, where basically we can fine-tune the initial hits by designing different warheads, or we can also play around with differences in linker regions. And since we already have a structure of ternary complex in place, we can also use it for more structure-based hit expansion or lead optimization. Yeah, the same is true for lead optimization. And when it comes to the methodology itself, there is a nice paper from 2019 that outlines 4 different methods or 4 different approaches of how to predict ternary complexes. So I'm going to introduce each of them into a closer detail. So in the first method, what we see is that we start with an entire complex where the protein of interest and the E3 ligase are connected to each other by the PIC compound, which has an extended conformation at the beginning. And then what the method does is that we evaluate all rotatable bonds within the linker region, and then we gradually sample all possible configurations. So, of course, there can be many problems connected to this approach, such as, for example, that we can introduce some protein-ligand and protein-protein clashes, because we basically start with a randomly chosen conformation. And it can also increase the time, because we basically start with the entire complex, which can be quite computationally expensive. In the second method, what we do is that we sample PROTAC conformations separately, and then we add a protein of interest and a given ethyl ligase at the very end, according to the fragments of the PIC compound. But again, we can run into some issues regarding sterical clashes. The third method is just a modification of the second one, where we basically take the PIC compound and we sample different conformations in the context of one of the proteins. So we mimic the scenario where the compound first bound to the first protein and then it attracts the second protein. And in the fourth method, we do protein-protein docking with the two proteins, and at the same time, we look at different conformations of the PIC compound, and then we combine it into ternary complexes. So what we do at Celeris is the modification of the fourth method. So this is the pipeline, a very high-level overview of the pipeline that starts with basically finding multiple protein structures per a given ethyl ligase and a given protein of interest. So in other words, we need to generate protein-ligand interactions with the two fragments. Then we do an exhaustive protein-protein interaction prediction with all different conformers of the POI and the ethyl ligase. And in the end, we insert a PIC or a linker inside of the protein-protein complex. We run a short optimization, and then we rank the compounds according to the energy of ternary complex formation. So when it comes to conformational ensemble generation, which is the very first step of the pipeline, as I said, we basically come up with an ensemble of different protein of interest conformations and the ethyl ligase conformations. We typically use around 10 to 20 distinct structures per given protein, and then basically the number of PPR runs equals to all the different combinations of POI and E3 conformers. And when we look more closely on what we need to do to generate such a conformational ensemble, we can either use X-ray or NMR or cryo-EM structures from the PDB, but we have to make sure that we know which of the proteins, which of the domains we want to target, and whether we can just model the isolated domain or we need to model the entire protein. Then also we often encounter that there are some point mutations, so we have to make sure that all the protein structures from the ensemble have the exact sequence. We also usually remove some excessive loops that are not relevant for ligand binding, and then we have to cap the structures. In case we do not have experimentally determined structures for the POI and ethyl ligase, we can use some ab initio approaches, such as, for example, molecular dynamics or normal mode analysis, or the combination of both. Okay, so this is just a bit more detailed slide about how we prepare binary inputs for the TCP pipeline, so I don't have to go through it. I just want to mention here that when it comes to protein ligand structure preparation, we typically start with already co-crystallized ligands, and we do some modifications on already existing ligands. But in case we do not have the exact ligand co-crystallized, we perform rigid body docking, where we use a substructure of the co-crystallized ligand as a template. Or when we do not have any corresponding structure with a given ligand in hand, we also do ensemble docking, where we probe different protein conformations, and we have a set of active and inactive ligands to identify the binding site. And here is just an example of what are the inputs for the TCP pipeline. So it's a compound with a given linker, and then of course the structure of ethyl ligase with a corresponding fragment, and the same for POI. So, as I said, we have two different versions of ternary complex prediction pipeline, where the first one is more driven by AI, where we use Bayesian optimization to effectively sample all possible protein-protein configurations and for effective evaluation of the objective function. So what we do here is that there is, like, the configuration space of protein-protein interactions is quite vast. So what we need to do is that we want to evaluate the feasibility of adopting a given protein-protein interactions by objective function. And more technically, we do it in a way that we select a subset of these configurations, and we calculate two different scores or two different fitnesses, where one of them is called protein-protein interaction fitness, And the second one is called constraint fitness, which somehow incorporates the effect of a given linker and how it affects the protein-protein interactions. Because, for example, if the two fragments are opposite to each other, there is no way that they can be connected by the linker. So we only look at those protein-protein interactions where the two fragments are close enough so that the fragments can be connected by the linker. This is what we mean by constraint fitness. And we combine it into a combined scoring function, and we train a model to predict the objective function, and it helps us to select, in the next iteration, to select another subspace of all possible configurations and do it in an iterative way until we find a representative subset of protein-protein interactions that we then cluster, rank them, and we filter for final poses. So then we also choose a bit different approach where we put our focus on protein conformational flexibility, which is not really addressed in the previous approach. And here we have an ensemble of structures for which we do a classical protein-protein docking. And what we do here is that in order to mimic the presence of a linker, we add a short spacer to each of the fragments to further restrict the exploration of possible protein-protein poses. And then we filter the final poses for exit vector distance, meaning what is the distance between the two fragments. We also look at the protein-protein interaction score, and we cluster the proteins according to LRMSD to account for structural variety in the ensemble. And then we evaluate different conformations of the PIC compounds, and we align them into the protein-protein complex according to their fragments. And then, as I said, we run a short minimization in order to optimize the complexes. So in order to rank the compounds, we use MMGBSA method. And to start from the beginning, we first need to calculate partial charges. And then we run optimization of the ternary complexes, which is being done in 3 different steps. So we first energy minimize the structure, and then we run simulated annealing starting from 300 kelvins going down to 0 to sample all possible conformations. And then we calculate MMGBSA energies. And when you look at this plot that shows formation of a ternary complex, we typically start with 3 isolated components with protein of interest, E3 ligase, and the PIC molecule. Then there is a transition state with less and more stable binary complex, and then there is the full ternary complex. So we basically enumerate and calculate energy for all these scenarios. And then what we typically do is that we take the relative difference between the initial and final stage, which is what we call delta G complex, and which is what we use for ranking the different compounds, then are then selected for synthesis and for experimental validation. So as I said, there are certain issues when it comes to validation of such an approach, because I already said that there are just 22 experimentally determined structures, and they all come from X-ray measurements. And this is a nice example why this can be a problem, because in this paper, the authors did resolve a structure of ternary complex between BTK and CIAP1 ligase. And you can see that they basically crystallize the same structure 3 times in 3 different replicas. And when you look at the overlap or the alignment of the 3 structures, you can see that there is quite some variety when it comes to orientation of the CIAP1 ligase compared to BTK. So, which tells us that there is a certain structural variety, structural dynamics that plays a role in adopting ternary complexes, and that's why it can be quite challenging to use the structures for comparison with our predictions. So what we do instead, we compare affinities with the MMGBSA energies to validate our approaches. So talking more into detail about the validation, we can tackle validation from different perspectives. So when it comes to validating target selection, what we need to do is that we have to collect a list of non-degraded targets versus random targets that are used as negative data points. And then we run TCP pipeline to see whether we can see a clear difference in energies between non-degraded targets and the the random targets. Then for the second point, which is about target selection based on selectivity predictions, we need to come up with the pairs of protein where selectivity has been achieved versus, again, some random pairs that are used as true negatives. For calculating minimum or optimal linker length, we need to have a congeneric SAR series with the same warhead, same E3 binder, and differences in linker region or linker size. When we want to decide which compounds we want to synthesize, depending on feasibility to adopt a ternary complex, of course, we also need to have a compound series, the more the better. For deciding which compounds to synthesize based on selectivity, we would need to come up with the pairs where we have the compounds for the same E3 ligase but different POIs. And then if we would like to, in the future, do some kind of structural predictions, of course, we would need to have more experimental determined structures to compare them with our predicted structures. So here are just some examples from the literature. So on this plot, you can see the number of papers with ternary complex predictions and how it evolved over time. So you can see there is not a lot. So this is still a niche field, but we are getting better over time. And here are just some examples of how people validated these kind of approaches in the literature. So most of the time, they basically had a series of compounds, and they did predict ternary complexes, and then they looked at whether the predicted ternary complexes can be formed or not. So they had more classification approaches, typically with 10 to 20 compounds or 15 compounds. There are also some papers where the authors did structure-based comparisons, and also some of the papers did correlation validation, where basically they plotted DC50 or ternary KD values with respect to energies of ternary complex formation as a proxy for, or as a mean to validate the method. So here are just some examples of the validation studies we did internally. So here is a data set for tyrosine protein kinase BTK in the complex with the CRBN ligase. And you can see that basically the authors did design a series of compounds with the same warhead, same E3 binder, and differences in linker length. And they saw that the ternary complex formation is basically triggered by increasing linker length. And on this plot, you can see delta H2, which is the delta which is the energy of the full ternary complex. And here on the y-axis, we have a parameter that we called number of successful linker insertions, which is something like, or which can be interpreted as number of poses where we could have inserted a linker or the entire PIC compound as a very crude measure for assessing whether a ternary complex can be formed or not. And we tried to reproduce the experimental data. And you can see that basically we were successful because with increasing or with decreasing energy of ternary complex formation, we basically were able to reproduce the trends that were shown experimentally. And then there is another validation study that we did in-house for BRD4 protein, which is most commonly used protein for designing PIC compounds so far. And here we just have 4 data points and more subtle differences in dissociation constants, where basically we can see that MZ4 compound was a slightly less effective PIC compared to the previous 3. And again, we can see that we were able to reproduce this computationally. So then there is a qualitative difference in the energy of ternary complex formation between MZ4 and the remaining 3 compounds. So here are just some examples to give you a bit of idea about the energies. So here we had an internal target that I can't disclose, and we had a couple of positive and negative controls. And we were able to see that when we look at different parameters, such as number of successful linker insertions, we can see there is a clear difference between the positive control and the negative control. And the same is true for, again, delta H of ternary complex formation. And basically, the protein was composed of 2 domains, and we were able to reproduce the same trends for both of the domains. And here is just some example to give you a bit of idea about the number of TCP runs that we usually run internally. So for one of the targets, we were at the stage of lead optimization. So there were 80 analogs of the initial hit that were designed by our medicinal chemistry department. And then we run 40 selected compounds through the TCP pipeline. And then finally, we selected 17 compounds for synthesis based on the, again, energy of ternary complex formation. And here are just, yes?
I'd like to ask, what are the axes on the previous graph, the DDE2, DDE3?
Yes, so it's just another basically label for the same. So delta H3 is the energy of full ternary complex, and delta H2, or E2, sorry, is the energy of more stable binary complex. And when we go back to this picture, what we mean by the energy of more stable binary complex is this one, is this stage, which was found to be also a good parameter for reproducing experimental data. And we also found that these two energy differentials, the delta H3 and delta H2, are highly correlated. So we most of the time use delta H3, but sometimes we also plot delta H2. So these are basically interchangeable. Yes. And here are just overall validation results where, again, we showed that basically we can reproduce, or we can classify the compounds into active, intermediate, and inactive when we look at our energy differentials. Okay, when it comes to linker generation, we have an in-house generative AI model that is based on a neural network. And the model works in a way that it takes the two fragments as inputs, alongside with different criteria that our medicinal chemists determine, or they want to design the molecules with, such as, for example, number of hydrogen bond donors, desired linker length, number of rotatable bonds, and so on. And the model is based on encoder-decoder architecture. It's trained on the drug-like subset of Campbell database, and then is transfer learned or retrained by using our proprietary repository of degrader-like structures. And the generation of linker itself works on the basis of reinforcement learning, meaning that there is an agent, which is sort of a tool that proposes a given linker according to the input criteria. And then the agent gets a feedback about its performance, meaning that there is a scoring function that evaluates different metrics, such as different Phys-Chem properties, linker length, rigidity, structural diversity, synthesizability. And then we update an agent and we repeat it as long as we reach convergence and we reach our desired linkers, which is an output of this model. So when we compile data to train the model, we have applied data mining approaches where we basically first fetched or gathered all the available PIC-like structures by running substructure screens, where we used E3 recruiter fragments as our substructures in order to identify compounds where this structure is a substructure of the overall molecule. But because we combine data from different sources, we have to standardize them before we merge them. So this is what we did by using our in-house data standardization protocol that works in a way that it strips off the salts and other fragments. It neutralizes the charges if needed. Then also we look at the drug-like entries on the entries with drug-like elements. And then once we proceed through the standardization protocol, we generate INCHI key, and SMILES that we then use for merging the data. And then we also look at some filters, or we use filters to filter for PROTAC-like molecules. So there are multiple filters that are based on given molecular weight, hydrogen bond donors, ratios of topological polar surface area, molecular weight, and C-log-P to basically reach the chemical space of degrader-like structures. And then, as a very last stage, we also do calculate Tanimoto similarity with PROTAC-DB, which is so far the most comprehensive data set of degrader-like structures. And here I'm just showing an example of numbers of degrader-like structures that we found in different sources. So it was quite surprising to see that PubChem was the most comprehensive source of degrader-like structures. And what I also want to show here is that when we look at the overlap, there is exactly 0 structures that would be found in all the sources, which basically approves our decision to combine data from different sources in order to increase our training data set. And this is what we combine with the in-house data that we measure in-house to then train the linker generator model. So, of course, there were a lot of challenges, especially at the beginning when we were developing the model. So I want to show here that we sometimes got a very exotic structures coming from the linker generator tool, where on one hand we had linkers that were very creative and novel, but not very stable. And on the other hand, there were a lot of linkers that would appear to be stable and synthesizable, but again, like they were not very innovative, and therefore it would be very difficult to patent these compounds. So what we did is that we basically used a subset of commercial building blocks that are used for developing PIC-like structures. And we used them to bias linker generation. So then we got structures with spiroatom groups, which are known to rigidify the linker and enforce certain conformation that may appear important for adopting a ternary complex. So this is just an example how we can modify a linker generator to bias it towards a given chemical subspace to optimize our linker generation. Okay, so when we conclude our efforts, what we can say is that the development of proximity-inducing compounds is definitely can actually benefit from including different in silico approaches. And what I've shown here is that we use prediction of ternary complexes that we can use for both structure-based design of proximity-inducing compounds and also for ranking the individual compounds to rationally select those that come to synthesis and to experimental testing. And I also talked about validation as a major challenge of the entire approach. Since we don't want to use structural experimentally determined structures given their limited number, so we usually use ternary dissociation constants or other endpoints from experiments to correlate our energies of ternary complexes with. So for linker generation, what we can say is that it can be useful for guiding our medicinal chemists to design structurally different linker to systematically optimize different compound properties. And I also shown that by using data mining, we can increase our training data set that is useful for developing different kinds of AI models, like our linker generation model. And then, in the end, by combining ternary complex predictions with linker generation is advantageous to drive design of proximity-inducing compounds. And here is just an illustrative workflow that shows how we can combine the two methods together. So what we typically do is that we have a series or a pair of E3 binder and the warhead, and then we exhaustively generate different kinds of linkers that we also evaluate for their physicochemical properties by running solubility and permeability models. Then we predict ternary complexes and we select a subset of compounds that make it to the experimental screening. And this is what we also do iteratively in order to optimize the compounds. Okay, and in the very last part of my talk, I'd like to focus a bit more on how is it like to...
May I just
yes?
have a quick question to the previous slide?
Sure.
You are saying that you are making the physical chemical property prediction that's done for whole molecule or only for the linker, and then you are basically adding those predicted ones?
Yeah, so when it comes to solubility permeability models, we use it for the entire molecule. But we can also use it as a post filter after linker generation to see whether the molecules should be retained or not. But it's done on the same on the entire molecule with all the three parts in place. Are there some other questions regarding the ternary complex predictions or linker generations? Or should we leave it after at the end? Okay. Okay, perfect. So I'm going to be very fast in the last part, which is basically about my thoughts and my experience with moving from academia to more industrial settings as a computational chemist. And I just decided to show this picture because I think it nicely illustrates the difference between academic and industrial work style, where basically when we look at the workplace, what I would say that in academia, even though it fluctuates, it's quite predictable because it usually aligns with academic year, where, for example, there is a peak when we need to submit a paper or when we need to submit a thesis at the end of the semester. While in industry it's more rapid, it's more gas break style, so the peaks are much more sharp, and it's always determined by different priorities in terms of the internal projects or technology developments. Then also what is different is the final product or deliverable, where in the end, in academia, it's mostly about publishing papers, sometimes also about patenting stuff. While in industry, it's basically about making the product to the market or some service, or we can also have different products that don't have to make it through all the clinical trials, but we can just stop at the preclinical phase and then out-license it to different companies. When it comes to time frame, I would say that academia is more, has more long-term projects, while in industry we often have to deal with very tight deadlines and very well-determined project timelines. Also, I noticed that there is a difference between the team culture, meaning that in academia it's more about working on the project individually, although we have academic collaborations, but in the end we are the ones who is on the paper. While in industry we work across different teams, and there is a very frequent coordination, meaning that even though like we collaborate with some external company, we have weekly or bi-weekly meetings where we have to give an update on our progress. And also, when it comes to decision-making, I would say that academia has a rather high degree of autonomy, so we basically are the project owners, and we can, to a certain extent, decide what to do with the project. While in industry, it can be, or it is usually quite hierarchical, and even in the small biotech companies like ours, which claims to be flat, in the end there is one person or two persons that make the final decision. And what I also needed to learn is that you sometimes have to get personally detached from a certain project, because even though like you work on a project as a computational chemist and the computations looks great, there can be some other reasons why the certain project can be stopped, such as, for example, due to safety reasons or due to problems with optimizing the molecules. So you can't actually take it very personally because you work on something and then in 3 months you can just the project is dropped and it's not basically your fault. So this is something that took me some time to learn.
Question on the previous slide, if I may.
Yes.
How do the areas under curves compare?
Yeah, that's a good question. I would say that, in the end it may be the same, but it's more about like how the work pays or how the work is distributed over time, right? So what I wanted to show here is that in academia, although we have some tight deadlines from time to time, it's much more equally distributed over the year, while in industry it really fluctuates a lot. Yeah. So then a lot of people ask me like how to make the switch from academia to industry and what skills you can actually take with you and what is important to highlight during the job interview processes. So of course we as academics publish papers, which is a kind of communication of how we communicate the results to our scientific community. So this is, of course, very much needed in industry as well. But in industry, it's not about publishing papers, but more about being able to do some advanced data visualization and clear communication to your colleagues or also across different departments. And what also helps a lot is to get involved in different public outreach activities when you are in academia, such as, for example, Researchers' Night, because oftentimes you also have to communicate with different people that are outside of the field of expertise, such as investors or some collaboration partners. They do not have a very clear idea about computational approaches. So it helps to also be able to communicate with non-experts, so to say. And this is what you can learn in academia by getting involved in different kinds of workshops or events where you present your results to the public audience. Also, when it comes to computational chemistry, I would say that in academia we tend to write a code that is very customized, and it may not be reproducible for other projects, which can be a problem in industry because it's all about having a code very well documented, to be also used by different colleagues or also people from different departments. And oftentimes we move from writing a code to using more data pipelining tools such as KNIME or Live Design because some of the colleagues are not that very well skilled in computations or encoding. So we stick to the pipelines that are more user-friendly, especially when we talk with people like medicinal chemists or biologists. Of course, as a PhD student, you teach a lot, and this is something that you can make use of when moving to industry, because you often work in different so-called cross-functional project team when there is one person for CADD or for computational chemistry, one person for medicinal chemistry, one person for biology. So you have to very clearly communicate and explain how your work or how your method works, also for the people that are not from your direct field. Also, when you happen to co-supervise or supervise a student, this can be very useful when you get to the more leadership role where you manage a team or you supervise some junior team members or research interns. And of course, like, this is a very, I would say, generic skill, but it's very important to mention it here because it's all about critical thinking, and this is what you learn in academia, and then in industry you can use it in a way that you can think of the task and you can just break it down into small manageable steps and then to delegate it to someone else because time is money, right? So the more efficient we are in doing some task, the better. And this is also something that yeah, comes with critical thinking and problem-solving skills. And here I just wanted to give you an idea of how does it look like to go through the job interview process. So we have recently, it was some months ago, opened a role for senior and principal computational chemist at either of our 2 locations. And I just wanted to give you, let's say, an estimate of how many applicants basically apply for this position and how many make it through the different stages of the interview. So at the very beginning, there is a call with the team lead or with the head of department, which is basically about introduction from both sides, talking a bit about the background of the applicant and the motivation to apply to our company. We also talk about expected salary and also the start date, which is very important for industry because yeah, when there is a project and there is a position that has to be filled, it has to be done as soon as possible. And at the very beginning, we had around 50 applicants. Then we selected 10. Then went to the second stage of the interview, which was about programming exercise, where we typically have a task of writing a Python script to solve some issues with protein structures or to measure or to analyze the protein structures in different ways. And then we select 3 top applicants that make it to the next round of interview, which we call structural bioinformatics session, where basically the applicant meets the entire team, and there are different questions related to molecular modeling, and it's more about figuring out whether the applicant can think of different solutions for different problems that we tackle in the company. So it's more conceptual, and we also look at whether the applicant would be suitable from the cultural perspective, whether it would be a nice team member who would be able to communicate with others. And during the same stage, we also involve the people from other departments just to understand whether the applicant would be able to talk across different teams. So we also ask him or her some basic questions from different areas such as medicinal chemistry, biology, or synthetic chemistry. And although we all know that computational chemistry is quite an interdisciplinary field on its own, I would say that there are 2 major roles or 2 major types of computational chemists in industry, where on one hand we have someone who's more on the applied side, so I call him computer-aided drug designer, which is a person that works on the project together with other teams, and which basically where the person would need to use different kinds of molecular modeling software in order to tackle different biological tasks and be involved in drug development process, which comes also with a good knowledge in structural biology, and the person also has to be exposed to medicinal chemistry to a certain extent to understand or to help the chemist design some changes or to optimize some hits, or to expand the hits. It's good to know to have some like chemistry, medicinal chemistry background. And then there is a second class of chemists, computational chemists, I would say, which are more focused on technology development, where, of course, coding skills are essential, and we mostly use Python and also some libraries like RDKit, because it's very handy when we work with chemical structures. It's also good to have a good understanding and practical skills in machine learning and different data pipelining tools. And that's it from my side, so if there are some questions, I'm very happy to answer them. Or if you have some comments or thoughts on targeted protein degradation, I'm very happy to talk it through.
Yeah, thank you a lot. So questions on both parts, the first one, the second one. Yeah, please.
Thanks for the great talk. It's really nice to see how industry works. And it's very beautiful work. But I have a question about the second part, especially about the industry jobs. So I was wondering, like, I did a PhD in the experimental, basically it was a wet lab PhD. I was wondering, how would you say, is it possible to transition from the wet lab experience to become a computational chemist or computational biologist?
And just to understand, you don't have any prior knowledge of computational chemistry. Are you able to use tools like Maestro or Autodoc-Vina, or are you starting from scratch?
No, I'm not starting from scratch. I did some structural biology and some work with, again, with Python scripting and with, not with Maestro, but with Chronos, but in general, like if you don't start from scratch.
I think there are very high chances to succeed, because we also have now people that are named as computational medicinal chemists, which are basically people with experimental background in medicinal chemistry, then transition to computational chemistry later in their career. So I would, as I said here in this slide, it's not crucial to know like programming to like very into a detail. As long as you can work with Maestro, you can be somewhere in between computational chemistry and medicinal chemistry departments. So these roles are also available nowadays, and they are very desirable.
So maybe the question, I'll have a similar question. The programming, as you said, there is a lot of coding. Is it more focused on actual coding, or is it more crucial for applicants to know general coding, or know the background of a method? Or it's a synthesis of both?
So in the end, what, for example, our pipeline is about is that we don't do everything from scratch, meaning that we sometimes use an existing tool and we just incorporate it in the pipeline. So
like the Unix philosophy, you just...
Exactly. So it's about also being creative in solving your exercise, but I would say it's more about knowing how to use it, you don't necessarily need to know all the like technical details and, for example, optimization of algorithms and these kind of things, because it's in the end like the pipeline is to some extent about using existing tools and incorporating them and combining them with others within a unified framework. So like you need to stick it together by writing some code, but you don't have to, for example, write a code for protein-protein interaction prediction from scratch.
So like Unix philosophy, when you write just a simple code, some simple little program to do some little task and then combine it.
Exactly. Yeah, this is how we usually work. Yeah.
Just to follow up regarding the open source, how frequently do you use open source? How frequently you contribute to open source?
Yes. Yeah, that's a good question, because at the beginning we started with using mostly open source software, like I can say, for example, Autodoc-Vina, but then we encountered that there are a lot of challenges because once the atom is named a bit differently, it just fails. And that's why we decided to stick to Maestro, because it's also more feasible for our colleagues that are not that very computationally skilled. We are slowly dropping from using open source. But again, like as I said, for example, KNIME is an open source data pipelining tool that we use very frequently, so it really depends. But when it comes to molecular modeling, we decided to move more to Maestro type of software. Yeah. Yeah.
You know, you used the model of promoting protein degradation via ubiquitin link. But what, is it possible to get some similar approach to more complicated cellular processes like membrane trafficking, like interference? Do you know, is anybody doing like interference with membrane trafficking proteins and
like
that kind?
Like for example, you mean by modeling lysosomal pathway?
Yeah, yeah.
By like
modeling and then synthesizing compounds that would interfere with protein-protein interactions during the membrane trafficking.
Yeah, I would say it's theoretically feasible to model, but then it scales up and it increases the computational time. So we try to facilitate the problem to only resolving ternary complex computationally rather than trying to model the entire pathway or some other cellular systems that are interconnected to targeted protein degradation. But of course, the pipeline is general enough to be applicable, for example, for the membrane proteins, once we have a good understanding of their structure. But it's not feasible to model, for example, lysosomal trafficking or endogenous uptake.
So for lysosome it's not applicable, no?
Again, like if there is a protein that is subjected to lysosomal type of degradation, we would model again just the ternary complex, but not the surrounding lysosome, and we wouldn't probably care about how it goes into the lysosome because it's not. It could be a project on its own, and it's not suited for industry, right? Because it would require a lot of time and effort. So.
Yeah, thank you.
I would like to ask if it's not secret, which diseases are you currently targeting in your ProTech development?
Yes, well, that's a good question because we can't disclose the target names, but I can say that we have a couple of programs focused on oncology diseases and neurodegenerative diseases. Yeah.
So, I just graduated medicinal chemistry on a bachelor level, and I would like to ask you, what are the 3 bullet points that I could work on for the perfect CD for the computational chemist position, for an example, in such a company?
Yeah, so some of our colleagues were basically after graduating in their masters. So we also accept more junior scientists in this respect. But if you are interested in how to proceed with your studies after bachelor in order to apply for such a position, I would say it depends. You said you are a medicinal chemist, right?
Yeah, bachelor level right now.
Yeah, bachelor level. So then if you would like to get more into computational medicinal chemistry, I would recommend you to do your master thesis in more computational chemistry based project to get your hands on different tools like Maestro or like binding site predictions and basically the methods that are taught at this workshop. So, and a bit of coding skills, like to have some Python courses or KNIME courses. And as I said, we also used to have colleagues that came to us straight after their masters. So we are not only focused on those that are post-PhDs or postdocs. Yeah, so. Welcome.
Some other questions?
I would like to ask, in the protein-protein interaction calculations, you were mentioning conformational sampling.
Yes.
I would like to ask that you said that there are some already experimental conformations from X-ray and other stuff, and then there are computational ones. But in the computational methods, there is a lot of degrees of freedom, like how many conformations you sample, and how much do they differ? Yeah, so can you provide some insight on how
Yes.
much, how extensively?
Yeah, so if we would like to generate conformations computationally, we just run a spontaneous MD simulation for about a couple of nanoseconds, and then we do a cluster, or then we do, I forgot the name of it, but basically the principal component analysis to pick up the most representative structures. But again, because we do ensemble docking, where we basically have, where we dock each POI conformer with each E3 ligase, we only limit ourselves to like 10 to 20 structures, because then it would again be very computationally expensive. Yeah, so.
Maybe when you speak about computational resources, how is like the private sphere in the industry in that matter, since universities have connection to maybe supercomputers? How does the industry stand? Do they have like their own resources, or they use some virtual resources?
So originally we had access to Google Cloud, so we were running our calculations in there, and now we stick to using our in-house machines that are physically like based in our offices in Graz, and when needed we also like purchase some virtual machines for a given time frame when we need to like get more resources to run our calculations through. So yeah, it varies. Like it's a combination of physical resources that we have in place, and we also sometimes purchase access to some virtual machines.
CPU or GPU?
So yeah, it's actually both, depending on which stage of the pipeline. Yeah.
Yeah, thank you. Sorry for that. Another like business-related question: do you mostly do your own product development, or do you, let's say, do you do like research for, let's say, larger pharma companies? What is your planning?
Yes, so our primary business model is to develop our drugs to the clinical stage, but as Professor Berka said, it can take up to 10 to 15 years to get the drug through all the clinical stages. So right now we are funded by different investors' funds, but we also collaborate with different external companies such as Boehringer Ingelheim or Merck, which basically pay us for doing the projects for them. So this is the way we can generate revenue. So the primary goal is to have our own drug on the market, but at the same time we are using our pipeline and collaborate with different pharma and biotech companies too. Yeah.
Please.
I have a question about job interview. There is this coding exercise, and I would like to ask if it's like you are sitting at the computer and you can code the way you want, or it's more controlled environment where I should code more from my head, what I remember, how it works actually.
So we usually give the applicants about half a day and to do the exercise on their own, and then we let them to present the results, to go through the code and to explain why they did certain steps like they did. So we leave them to do it on their own. But for the last interview, we also had like a website or a platform where we can actually control and see how they changed the code over time. So how they went back and rewrite something to just better understand like what is their process of thinking, to yeah.
Thank you. I apologize. It's really tricky. What if they admit that they used some language model to generate the code instead of writing it, and the language model did it correctly? So it's not their own code, but they knew how to generate it efficiently.
I would say it's not the problem on its own, as long as they can very well explain what the individual stages do. Because also in reality, when you work in the company, you often do Googling in order to look for some information. So we don't want to forbid this kind of approach, but even though like the code would be partially generated, they have to have a very good understanding what it does. So that's the most important and crucial part.
Can you use this code which was generated, like some language model, just with this some IP issues, or you can use it?
Oh.
Generate some part of the code with just GPT. Can you use it in your development?
I mean, not in the development, of course, but I think you were talking about the job interview process, right? That, yeah. So of course, like, yeah, for the IP issues, yeah, everything needs to be written in-house. Yeah.
That's your question regarding this AI-assisted linker design.
Yes.
What was the major novelty come from your development, from linker or from this part of both ends of the linker?
So your question is about whether we focus more on modifications in the link region or the warhead?
If you want to patent this compound, you can modify either linker or those two heads, warhead or another part.
Yes. So we typically focus on modifying the linker region because, and we want to already start with existing inhibitors because it's easier to develop the.
Major novelty comes from linker.
Yes. At this stage, yes. I mean, of course, in the future, we want to move to also finding novel warheads, but it adds another layer of complexity to the overall development process. And we are a very young company, so we start from already existing warheads and existing binders, or we do some very tiny modifications, but we focus on fine-tuning and optimizing and designing the linker regions.
How many building blocks you use for this linker design? Just roughly?
I would say it was around 100, yeah.
But in this case, I'm just curious, why do you use this AI technology with autoencoders to generate these linkers if you are still limited with a quite small number of building blocks? You can enumerate all of them exhaustively and test.
Yeah, but then we had the issues with this, right? So sometimes when we didn't bias the generation towards specific subset of chemical space, we ended up with linkers that are like chemically very very like unstable and not very usable.
Yeah, that's what I'm asking. So you can enumerate exhaustively all these compounds with all your building blocks. You don't need to generate from scratch like these autoencoders which you use if you have a limited number of these linkers. So I just want to understand what's the benefit from this AI part.
So it's about basically using multiple different criteria at the beginning. So it should basically ease medicinal chemists their job because they would otherwise draw the structures on the paper, right? And here we can just specify which all different criteria we want to have when we design the linkers. And then we basically get the linkers, and then they are anyway subjected by visual inspection from the chemist to select a final subset. So we typically generate around 20,000 different linkers or 20,000 different molecules. And then we also apply different kinds of filters and visual inspection to shrink it down to, let's say, I don't know, 200 molecules then that we then use for ternary complex predictions. Yeah.
Please.
So I'm curious about the third part of your molecule, which is native linker, not warhead. The term E3 ligase is run up multiple times across the presentation. I'm not exactly familiar with that. I was wondering, is that a constant among these proximity-inducing compounds, or is there also a variety or a library of these? Either the small molecule anchor, what do you call it, or is there a plurality of these ligases or proteases to bind to?
Yeah, so I would say currently there are around 20 E3 ligases that are being used for developing degrader-based drugs. And they often come with very well-defined scaffold. So there are also modifications to the scaffold, but there is not so much variety, I would say. Like, for example, CRBN always come with polydamyloid-based scaffold. And yeah. And there are modifications, but usually the scaffold remains the same, and we want to stick to that scaffold. So then when it comes to some variations, we more focused, as I said, on the linkers or eventually on the warhead that binds to the protein of interest, where we can look for more structurally different scaffolds.
Some other questions? No. I think that's all for today. So thank you once more. Now, the coffee break time and the poster session time, so just refresh yourself, come to the poster, just to discuss with your colleagues.
It's not the end, actually.
We are too late. 40 minutes.
It's a yes or no question.
A teď musí být trochu